Yes, now it's part of
So, the lamp lighting ceremony now to be streamlined. In the presence of the dignitaries, the holy lamp would be lighted, and along with that, there will be Saraswati Vandana, Saraswati, the goddess of learning, blessing us for the successful execution. I welcome all the dignitaries from San Pedro College also to join us virtually in this lamp lighting ceremony. We are witnessing the lamp lighting ceremony of this international e-conference. Professor Rajkumari Ghosh now lighting the lamp. This is Dr. Ashwini Kumar, Assistant Dean, Department of Engineering. We are confident that Sister Ida T. Frankilo and Dr. Arvind Fowler have joined us in this lamp lighting ceremony.
என்ன டிஸ்கஸ் பண்ண போறீங்க? நேற்று இதில் பாலாஜி பாலாஜி கால் பண்ணியிருந்தாங்க. டாக் வித் பாலாஜி என்ன கேட்டேன்னா இந்த விஷயம் தான் இவரும் கால் பண்ணியிருந்தாரு. மிஸ் கால் சேம் பாயிண்ட் தான் சார், நான் சொல்லிடுறேன். அப்புறம் உங்களை பேசுறேன். சொன்னாரா?
Witnessing the lamp lighting ceremony and getting blessing from Goddess Saraswati, we will move ahead for a condolence on the sad demise of Dr. Balkishan Razda, Professor of Birla Institute of Technology, Mesra, India, and who was also an author of repute. We will request all the dignitaries to stand up for a minute and put our condolence on the sad demise of Professor Dr. Balkishan Razda. Thank you. Now, in this inaugural session, let me invite Mr. Jasbir Singh Dhanjal, Registrar, Arkajan University, to put forward his ideas on this particular conference and enlighten us through his words of wisdom. Mr. Jasbir Singh Dhanjal. Let me invite
a very good morning to one and all present in this e-conference from Arkajan University and along the globe, San Pedro, Philippines School of Pharmacy from there, and to all delegates and distinguished speakers. It is the first step which we have taken through the MOU signed with this San Pedro College, Philippines. For this MOU was signed between School of Pharmacy, Arkajan University, and I really appreciate the step taken by Dr. Sau and his team, basically to give this e-conference where we can learn from the speakers who would be for next 2 days enlighten us with their research work and probably the theories, what we come to know, and rightly the topics is most demanded of this generation. Natural, when we talk about natural, people are basically attracted towards it. The topic natural novel pharmaceutical research will basically help us to get the past which we have lost. And definitely with this past, we are going to be benefited and fight among the diseases as we are coming towards this lightened globe. And with my well wishes to the Department of School of Pharmacy and St. Frederick, Philippines for this jointly conducting on this topic and helping for the listeners to enlighten their knowledge. And thank you very much.
Thank you, sir. You have simply put the words on the nerve. That is the theme of the conference. Now, there will be an audio clip address from Sister Adia Di Franquillo, President, San Pedro College, the Philippines, who is also the chief patron of this conference. I would like to request the technical assistants to play the audio of her talk.
Professor Dr. Razi, Vice Chancellor of Arkajan University, India, Assistant Professor Fatima May Tesoro, and Mr. Amit Srivastava, patrons. Dr. Jyotirmaya Sahu and Dr. Erwin Valer, conveners. Mr. Alok Kumar Muharana and Ms. Danielle Matuginas, organizing committee heads. Our event coordinators, Ms. Khushbu Raj and Ms. Yugita Kumari. Distinguished speakers, experts, researchers, faculty, and students. A congenial morning greeting from San Pedro College, Philippines. It is truly fascinating to know that two learning institutions, Arkajan University, India, with a School of Pharmacy, and San Pedro College's Pharmacy Department, were resolute in their decision to forge time, talent, and resources in order to realize this conference
पण I hope
titled
आलं.
Natural and Novel Pharmaceutical Research as the Key to Unlimited Discoveries. Surely, the organizers are with high hopes that academicians, scientists, pharmacists, and students participating in this two-day conference will be immensely
काहीतरी वायफायवर बोट
enriched
केलं सांगा
through the
पण ते
sharings
आता मला
from
माझ्या
renowned
लेक्चर वेळ तरी
speakers
आलं पाहिजे
at the
व्यवस्थित
center's
एकदा सुरू
investigative.
झाली की मग पुढचं सोपं होऊन जाईल
It is also my wish that your learnings from this conference may kindle ideas which will be translated into products to help alleviate human infirmities. I commend the efforts of everyone in creating this noteworthy engagement. On behalf of San Pedro College, I offer my warmest thanks to everyone who is present. May you find this virtual conference rewarding. Good day, everyone.
Thank you. And clappings from our end. So the audience might be listening to the clapping, the real clapping on virtual mode. However, thank you, ma'am. And you have truly said that this conference would certainly end up with some new explorations and will invite new talents to go for natural and novel products for pharmaceutical researches. Thank you so much. This was Sister Ida DiFrankilo, President, San Pedro College, Philippines. Now we will have an address from the patron cum Dean, San Pedro College, Professor Fatima May Tesoro. Ma'am.
Thank you so much. A pleasant morning to everyone. Professor Dr. Razi, Vice Chancellor of India, Sister Ida T. Francilio, President of San Pedro College, our chief patrons, my co-patron, Director of IQAC of Arca Jain, Mr. Amit Srivastav, conveners, we have Dr. Jyotirmayu Sahu and Dr. Erwin Faller, our local organizing committee head, Mr. Moharana and Ms. Matuginas, professors, pharmacists, student organizers, friends, benefactors and colleagues, Dominican greetings, mabuhay. I always ponder on a Chinese proverb that goes, when the winds of change blow, some people build walls, while others build windmills. I must say, the pandemic blew things out of proportion in such a massive scale. Do you agree?
Yeah, of course, ma'am.
Agrees. Yes, I can use all superlatives to describe the trajectory of declining social economic status that's across border. The impact of pandemic was really horrible. It blew us all in different directions. Even in the academic environment, we still continue to grapple. However, our resilient spirits allowed us to see through things differently. Hence, this international e-conference has come to play, let alone two universities converge to making this conference possible. It is because of the boldness of these two institutions that we were able to face the wind of change. The leadership of the pharmacy departments of Arca Jain and San Pedro College harnessed the use of the virtual platform to gather distinguished speakers and researchers and presenters so that we do not stop exploring natural and novel researches on drug discovery, on herbal medicines. And second, the idea of building windmills and not walls. This inspired us to reach out to as many possible institutions, share experiences, share best practices in utilizing knowledge and translating them into research. Hence, my profound happiness as we in San Pedro College enjoy all participants in this conference together with Arca Jain University to be fully engaged in the conference. Together, we'll listen and learn from our distinguished speakers and have fun in this academic pursuit. I fervently hope to see you all next year, soon, when we will have a face-to-face as we build more windmills together. Good morning, everyone.
Thank you so much, ma'am. Your talk had two important, remarkable, significant indications. And the first goes that Arca Jain University and San Pedro College are ready to face the wind of change. And the second is that we need to utilize the knowledge to translate into new products. So these two I found are the basic fundamental thoughts through which we should get united and hope for better tomorrow. Thank you so much, ma'am. Now, there will be an address by convener and dean, School of Pharmacy, Arca Jain University, Dr. Jyotirmay Sahu, who will be delivering on the thematic aspect of the conference and his personal experience with this conference. Dr. Jyotirmay Sahu.
Good morning, one and all, in the country and across. Namaste. A warm welcome to the International E-Conference, collaboratively organized by the Department of Pharmacy, San Pedro College, Philippines, and the School of Pharmacy, Arca Jain University, India. All the dignitaries and the participants from different parts of the globe, I am expressing my gratitude to all of you for your overwhelmed support. Professor Erwin, thank you very much for your kind cooperation to extend your hand to build this new friendship. There are a lot number of movies in India witnessing the friendship. If you will come, we will see it together. I can show you, and I would like to enjoy the movies like Sholay and Yarana from the Indian cinema, which witnessed the friendship. President Aida, Professor Erwin, Dean Madam Fatima May Tezro, Mr. Daniel, Professor Joseph, and other from San Pedro College, Philippines, we are delighted and quite speechless to express the happiness with the beginning of this friendship. A huge round of applause in the name of this friendship between India and Philippines. Thank you. I am thankful to Reverend Shuman Tiwari sir, Assistant Director Drugs at Jharkhand Drugs Control Directorate, Professor Dr. S.G. Deshpande sir, Ranjit Variskar sir, Kausik Desai sir, Dr. Krishna Priya ma'am, Dr. Ajay Semalti, Dr. Mona Semalti, Sharvan Kumar sir, Nagaraj Rao sir, Dr. Ajit Thakur, Bandana ma'am, Ambar Vyas sir, my friend Ajay Singh, Chitranjan Sahu, and Saktiketan Prushti, who are really inspired me to go for this international conference. And fortunately, I am also excited how it has been gone fruitful today. This is all due to your support. My teachers, particularly Professor Dr. D.K. Tripathi, Professor Sudhir Kumar Paide Sethi, Professor R.N. Padi, sir, without your blessings, this chapter may not possible. I would definitely like to acknowledge the support of my director, Honorable Mr. Amit Srivastavji, Vice Chancellor, Dr. S.S. Raji sir, Registrar sir, Mr. Jasbir Singh Dhanjal, Director of Campus, Dr. Angad Sai Tiwari sir, and Chief Financial Officer Rizab. Dear colleagues, and above all, dearest students, this would not be really such colorful without your coordination. And just would like to show you something in this regard. IT, please run the video. Thank you. Yes, the school belongs to Arkajain University, School of Pharmacy. Just under the soil, not yet getting suit up for last 3 years and waiting for its germination. Its nourishment and growth really needs your support. So I just need a drop of water and a handful soil to grow this organization, School of Pharmacy, in the upcoming days, which should be renowned across the globe. And this can be only possible with your kind support. Praying before God, the everlasting friendship with San Pedro, and also with all of you. Thank you very much. Concluding my speech, Dr. Jyoti. Thank you.
Thank you, sir. This was Dr. Jyotirmay Sahu, Dean, School of Pharmacy, Arkajan University. He notified that every step is important. We should not linger on any time. And the notation of deciding and acting. Yes, Arkajan University and San Pedro College have decided and started to acting upon the decision. And that's the fruition of this e-conference. Now, with great sense of privilege and honor, I welcome the Chief Patron of this conference and Honorable Vice Chancellor, Arkajan University, Professor Dr. Syed Saftar Razi, a renowned zoologist and expert of hormonal studies, who is with us now. We would like to offer a sapling on behalf of this conference to Professor Dr. S.S. Razi. May I request Dr. Jyotirmay Sahu sir to offer the sapling to Dr. Razi on behalf of Arkajan University, School of Pharmacy and San Pedro College, the Philippines. So you are witnessing the receipt of the sapling so that it grows and grows and shoots up to the sky. May I request Professor Dr. S.S. Razi to enlighten us with his words of wisdom on this particular occasion. Sir.
Well, good morning to all of you. It gives me immense pleasure to be amongst you all. I'm glad that Department of Pharmacy is doing very well in Arkajan University. They have organized various conferences, seminars, and workshops. As a matter of fact, research is one of the very important things in any field, whether it is in social sciences, or whether it is in technology, engineering, or whether it is in biohealth science. Well, as a matter of fact, the topic that you have chosen, pharmaceutical research, the very meaning of the research is that the more you go deep into the subject, the more you come to know about that. Well, as a matter of fact, there are two rates. We all know natality rate and mortality rate. Well, the mortality rate has gone down. Why the mortality rate has gone down? It is just because the health science has improved. And so the health of the mankind around the world has improved. Well, we are going through pandemic. There is no doubt in it that for the last about 1 and 8 months, we all in the world, whether it is European country or whether it is Asian country, we have suffered a lot, including our students. We have suffered the various deaths that has devastated the world due to this. But to some extent, we have overcome. Because you all know that there is mutation, a term mutation. What is that mutation? Mutation is nothing but it is a change. Now the change that takes place, whether it is a virus or whether it is bacteria, they know how to survive. We take medicines, we take vaccines, but they are also very clever. They make a shield around that in order to protect them. And that is why there is the pandemic phase 1, the pandemic 2, and we say that the pandemic 3 will come. Anyway, with this research that is going on, as I told you, the pharmaceutical research, that the mortality rate has gone down. Because various medicines that has come, including the antibiotic. Previously, there was one antibiotic, one type. Now there are various types of antibiotics.
They will be seeing me at least.
Hello. Well, I'm thankful to the research scholars and the guest speaker, those who are going to participate in this international conference. On behalf of Arka Jain University and Jain Group of Institution, I welcome the delegates and of course, the speakers, keynote speakers. Once again, I would like to pay my respect to them on behalf of our university. Hope this type of international conferences or seminars will continue so that the faculty members will enjoy, will learn, and then we will go further. With these few words, I'm thankful to Department of Pharmacy, all the faculty members of the Department of Pharmacy, Professor Jyotirmay Sahu, who is the Dean, and of course, the management of the university, the director of the university, Amit Shrivastava, the registrar, Professor Jasbir Dhanjal, Dean, School of Law, Assistant Dean, School of Engineering, and all my colleagues. Please continue these type of conferences, these type of seminars in other departments also. The other departments are also doing very well in Arka Jain University. Thanks.
So we were listening to Professor Dr. A.C. Razi, Honorable Vice Chancellor, Arka Jain University, India. Thank you so much, sir. Your regular inspiration and blessings with inherent quality of pouring affection to each and every faculty members of this university would certainly encourage us to go for further such conferences in frequent dimensions. Let me invite now the convener and director, Faculty of Pharmacy, San Pedro College, Dr. Arvind M. Fowler, for his address. May I request you, sir, to unmute and address the gathering.
Yes, thank you very much. Actually, our Dean of the School of Pharmacy here, Professor Fatima May Tesoro from San Pedro College is here, very active with us. We have also our President, Sister Aida T. Francilio, and our beloved students who are very, very enthusiastic to really learn with our international conference. They are actually in the YouTube live listening also. At the same time also, I would like to thank our partner from Arka Jain University, headed by the School of Pharmacy, Professor Sahu, and also with the Vice Chancellor, the Registrar, the guests, and of course, the organizing committees who are here with us, enthusiastically organizing this event. Madayaw kanatong tanan. Good afternoon to all of us. Now, first of all, I would like to acknowledge both parties with regards to organizing this event. From Arka Jain University, when Professor Sahu approached us with regards to co-organizing this event, we cannot actually say no, because in this advent of pandemic, this actually marked us the unity and of course the partnership that we have with our international partner from Arka Jain University. With this international gathering and conference that we have, this actually marks our looking forward for our face-to-face engagement for next year. And according to our Dean Fatima May Tesoro, she is actually very excited to visit Arka Jain University, India. And actually, the whole administrators of San Pedro College are very excited to be there in Arka Jain. And we would like actually to visit and benchmark in your university. We would like also to experience with our students, with our global mobility program, to really be there and listen to your professors with regards to, the quality and excellent education that you have in Arcadia in University. And in lieu with that also, we would like to accept your students to be with us in the Philippines in terms of mobility program, internship program, research, and etc. We have a lot of commonalities aside from all the differences that we have, but we actually moving forward to accept your students and faculty also here in the Philippines. In behalf of San Pedro College, we would like to convey our congratulatory remarks to Arcadia University and also with the School of Pharmacy headed by Dean Tesoro with this, and also with Arcadia in School of Pharmacy, Professor Saho, for this exciting initiative. And this is not only the first and the last, but this is actually the start of collaboration, not only with the conference, but also with research, mobility program, and etc. And this is actually, this international conference will be the showcase of researchers of both parties, and also invited lecturers, guests from both universities that will showcase its researches on natural product development and cosmetic product development. With this, thank you very much and congratulations. And we would like to welcome you all for this engagement of our international conference. Thank you very much. Daghang salamat po.
Thank you, Dr. Fowler. And we hope long live this friendship and collaboration. Now, we will have an address from the guest of honor of the day, Dr. S.G. Deshpande, former principal, CU Shah College of Pharmacy and ex-chairman, All India Board of Pharmacy Education. We are honored, sir, to invite you for your talk, Dr. S.G. Deshpande.
Good morning, everybody. Can you listen me?
Yes, sir, please.
Oh, good.
Yeah.
So I'm really happy to be in this group of scientists working in different areas of pharmacy from Arca Jain University as well as from San Pedro University of. This is the beginning of collaborative activities, and I think in future also you will do well. Now, when I heard Dr. Sahu, I realized that anything which has to grow properly will normally take a lot of time. And that has been seen from the development your college has been doing. It's nice. I'm very happy and I congratulate all of you for doing so well. Now, I must say one thing, that in India also, I mean, we all are concerned with the health of everybody. And if you look at the scenario, that everybody is trying their best to see that these healthcare products are made available to a common person. Now, when you look at the history of our ancestors working in this direction, I was happy to see Dr. Saud's plant growing. Our earlier generations worked for years together to come to certain conclusions regarding maintaining a good health. And those people have worked hard from generation to generation and come out with a technology which and the information, which of course is very useful to all of us. We must be happy that they could work for years together, generation to generation, and most of the rishis, our scientists during those days, we call them rishis only. They have seen the activities, and now they have come out with those basic things by which we can maintain our health. Now, it is already considered by them, after doing a lot of work in the health maintenance of health, that if you can control your food properly, your diet, your exercise, and mental peace, you will never suffer from any disease. Their purpose of work was to see that we maintain good health without having any disease. How diseases come, they have also given the reason for that. The moment you deviate from the normal course of activities, you suffer from diseases. And that is how Ayurveda has also come into the practice. And in India, for centuries together, these Ayurvedic people have gone to different forests, collected plants, and fortunately we are happy to have large number of plant kingdom, which are definitely helpful. But they wanted to really know which part of the plant is helpful, in what season the active principle is maximum in the plant, and collect them. And this was after getting this information, generation to generation, these Vaidyas, or so-called, experts in pharmacy, were diagnosing the diseases and controlling those diseases without any difficulty. This was a trend in India for many years, and they were maintaining good health because of good lifestyle, as well as in case they change the lifestyle to some extent, maybe because of some personal problem, there were people who would guide them by giving these herbal products, minerals, animal products, and societies are doing really well. But things have changed when we had the Industrial Revolution in Europe, and subsequently even in India. Earlier, it was only restricted to small towns and villages, and those Vaidyas were definitely helpful to take care of these people. Somehow, research is being done throughout the world, and certain diseases were not under control when, especially, these microbes were creating a problem, and probably that was the reason the immunity was reduced. Now they have realized that probably in 19th century, that we can control by preparing certain vaccines, and this has come from Europe, and they have helped a lot to maintain the health of people throughout the world. And this was Louis Pasteur, Jenner, and in their name, they have come out with those vaccines, which are definitely preventive medicines for these type of infections. Now, knowing their contribution, they called it as vaccinology. This term was coined by people during those days, and known that all these vaccines may be of good help to prevent many diseases. And fortunately, in India also, we are doing very well. So after that, in Europe, synthetic molecules started coming. They realized that some of these molecules, especially derived from the plants, instead of taking the plant product, you divide or separate the active principle from that and give it to the patient. and they could correct the disease to a larger extent. But after as these were coming after the Second World War, especially when we have developed in European countries only, the antibiotics, penicillin especially, which is a boon to our society, and these antibiotic along with sulfa cured large number of infectious diseases, and these diseases were very much under control, and slowly, naturally, these synthetic molecules started coming, and these molecules have certainly helped to cure many more diseases, because we had not gone little different from the normal way of life, and that's the reason these chemicals which were given in a formulation form, they were given to different people. Now these molecules, because we were under British Raj, came to India also, and some of the companies in India which were just in their infancy took the raw material from them, made some formulations, and these formulations were distributed in India to cure those diseases. Now, this was the scenario up to 1947. After 1947, when we got the freedom, we started some pharma industries in India also, starting from Bengal to Mumbai and then many other places. And these industries started growing with the encouragement from the government existing in India during those days. This has gone to such an extent that we are in a position to manufacture large number of pharmaceutical products. And this happened because many more pharmacy schools have come into the existence. These schools have produced experts who can definitely undertake the work of developing these products. And now many pharma industries are also doing very well. Today, India's position is third in the world so far as the volume is concerned. And so far as the generics are concerned, we should be proud. We are number one in the world. This is called as pharmacy of the world. And our people have been doing excellent work in different colleges, helping industry. And it's vice versa, of course. Industry and pharmacy colleges are exchanging experts. In collaboration, they are in a position to help a lot. And many of them are developing the formulation also, which are helpful. Now, in this connection, I must say one thing. Herbal products, of course, did their best. They are doing even today. Herbal products are given to individual patients. That's main thing we should remember. That is, it was a personalized drug delivery system by these experts who are having knowledge of all these herbs. We did new products, developed, an industrial revolution was responsible for it. That large number of industries came, large number of products were manufactured. And if you have some particular disease, then those tablets, those products, those capsules were given to different people. So there was no question of giving individualized based drug therapy. Now, ultimately, what happened because of this? Lifespan has gone up definitely with these new molecules which are coming in the market. But unfortunately, it's observed that the people suffer from many more diseases. That's because of the side effect. I have one thing from one Dr. Osler in UK made a statement. 1 minute. A patient who takes a modern medicine or allopathic medicine, as we call it, must recur twice from the problems. One is side effect of get rid of the modern medicines must recur twice, once from the disease and one from the side effect of the drug. Because side effects are quite high. And that's the reason, though lifespan has gone up, people suffered a lot. because of different diseases which are coming up because of these molecules which were there. You hear about diabetes, you hear about hypertension, you hear about cancer and all. These diseases increase and allergy too because of this industrialization, pollution problems which were not seen earlier. But today, all these problems are cropped up. And in this connection, there was a reference of one journal in USA where they are mentioned that many people are dying because of the side effects of the drug. They are afraid of taking these molecules. There was one open letter written to the President Bush by one Matthews Rath. I'll read that. He writes very correctly that on 9/11, 2,600 people, innocent people died in USA. Same number of people, same number, I'll repeat, same number of people are dying from the side effects of prescription drug in 10 days in USA. No, you can understand what must be happening in other countries. But that is going on. Now, of course, most of the scientists realized this problem. This was in 2002. And the Dr. Osler's statement that patient has to take recover twice, these are of the last century. This century, many more people have realized, many more colleges are coming up, many and experts are available nowadays in India also, and they have realized that the development can be done together by a team. And all these slowly people started knowing the importance of chemistry, pharmacology, pharmacognosy, and drug delivery systems. So knowing these problems, that side effects are there. They are prepared controlled dosage form, novel drug delivery systems. In fact, in India, we are also involved in doing all this work. We have developed many targeted drug delivery system by which the doses of the drug can be reduced and the side effect can also be reduced. That is the main purpose of all these things. And after biotechnology division, many of us realize that you can develop many more molecules. Gene therapy has come, genome is being talked, artificial intelligence is used in the development of pharmaceutical molecules. I mean, the synthesis of molecule. At the same time, you can have drug delivery systems. And with gene therapy, the scenario has changed to such an extent that we can have customized, personalized drug delivery system. Even people are working throughout the world. There is a collaboration with many people, Indians, and I'm very happy that Philippines and Indians are doing work in collaboration. Many more universities will come. So far as the development is concerned, all are required to get inside the formulation part, synthetic part, and develop those drugs which are safe. It is a dream of pharmaceutical people to have a drug which is free from side effects. Now, knowing that our country is headed by a very efficient prime minister, is encouraging, giving a lot of funds to develop products and inside the country, that people are getting the funds, working in different areas, and I'm sure that they will certainly try to reduce the toxicity of all these molecules by using the biotechnology area so that people get the benefit of all these molecules. Same time, now knowing that we are again thinking of personalized medicines as our ancestors were thinking, will come now with the gene therapy. Similarly, nanotechnologies also, I think Dr. Vandana Patel is doing a lot of work on nanotechnology for the last many years. And it is in collaboration with many other universities in the world. So I appeal to, this being a pharmacy week, uh, I appeal to all the pharmacists, experts, scientists, and budding pharmacists which are going to study now. They will have a bright future and they will get involved in development of work, distribution of work, medicines properly, and these medicines should go to the right people to get the benefit of these. I'm sure all will get involved in that, and I wish all of you good luck. Please continue working in this direction. Though I'm slightly away from little research because of age and all, but I'm interested in knowing what is going on, and I'm interested in working also with people, so that type of work is going on. All the best to all of you, especially Arka Jain University, the expert pharmacists from this college, Pedro College from Philippines, and I wish them that they work together to develop medicines and help people to get rid of the diseases. Thank you very much.
Thank you so much, sir.
Yes.
And the sound of the applaud might be going to you from the audience sitting over here. We are really enlightened today by the talk you have delivered to us. And my colleague, the friends who have joined us from the Philippines might have got the input that India is working hard on this path with a dedicated ministry called Ayush Mantralaya working on herbal medications and practices. Thank you so much, Dr. Deshpande. Now,
Thank
we
you.
are,
Thank you all. And all the best to all of you.
thank you so much, sir. Keep blessing. We have our chief guest, Mr. Sumant Kumar Tiwari, Assistant Director, Drugs at Jharkhand Drugs Control Directorate, Ranchi, India, will be delivering now his presidential talk of the conference in this inaugural session of the 3-day international e-conference on natural and novel pharmaceutical research. I request with warm regards Mr. Sumant Kumar Tiwari, our chief guest of the day, for his talk. Sir. Before Suman sir initiates, may I request our registrar sir, Mr. Jasbir Singh Dhanjal, to offer virtually our memento to Dr. Deshpande for his presence with us. Sir. The memento is being offered to Dr. S.G. Deshpande, our guest of honor.
Okay. Thank you very much and all the best.
We will be sending it to you, sir, later.
Okay.
And receive it now, virtually. Thank you so much.
Thank
Mr.
you.
Jasbir
Thank
Singh
you and
Dhanjal.
all the best.
Now, the address of the chief guest, Mr. Sumant Kumar Tiwari. Sir, please. Mr. Sumant Kumar Tiwari, sir, I think you can hear us. Please stay with us in this technical glitch, which is quite often in such e-conferences, but we will resort it out very soon. Mr. Sumant Kumar Tiwari, sir. Yeah, we are waiting for your address, sir. Please continue. Sir, you are not audible. Not yet, sir. You can remove the jack once and then put it once again so that it starts functioning. The jack pin of headphone, sir. We think there is a problem in that. And if it is wireless, then it's okay. But I think it needs to be reconnected. May we request to rejoin, sir? If you want, disconnect and join us once again. So you can opt the wireless headset option in the setting part of your laptop or PC. Suman, sir. Dear delegates, we are talking to our chief guest. He will join us very soon. Please stay tuned. So we can't hear yet.
हाँ हेलो। हाँ मैंने देखा यहाँ, हाँ लिखा हुआ है, मैं उसी पे हुआ अनम्यूट, उसको म्यूट।
सर आपकी आवाज आ रही है, प्लीज कंटिन्यू। वीडियो जरा सा ठीक कर लिया जाए सर।
हेलो, हाउ इट्स ओके?
Yeah, sir. Please adjust your camera, front camera. Yeah, on the screen your face is not there, so please adjust it.
Okay.
Yeah, place it towards you so that we can see it you on the screen. Yeah. This is fine now.
Yeah, thank you so much, Mr. ABHISHEK Ranjan. Good morning, ladies and gentlemen. Now we have come together from across the world for a novel in International E-Conference on Natural and Novel Pharmaceutical Research, collaborative, organized by one of the top college which is going to emerge in the Eastern India is Arka Jain University. In the very short period of time, their association with the pharmaceutical field, they have managed to organize such a big magnitude. Dr. Ranjit, and I'm not able to read out the name. So please, there's all knowledgeable person, Dr. Kaushik Desai, Ranjit Vashikar, and they all are all having a very eminent personality in the field of pharmaceutical education as well as in the field of research, academics. So it's my privilege. It was organized by the state of Jharkhand. I am the person who is welcoming all of you from across the globe. That is, this is the thing that we should feel proud on. And in this smaller period of time, in a post-corona era, the online, this thing, all the students, all those beneficiary, I have seen the participant across the globe, most of the participant from the western part of the globe. So it is, I welcome all of you, and I really pray that it's going to be a huge success for beneficiary of all students, all stakeholder, including the FDA people, including the community pharmacies, including the research which is been going on in the state of Jharkhand, eastern part of the country. Gentlemen, all of you know that is the western part and the northern part of the India is the pharmacy hub, manufacturing destination. In 20 countries. We have 1,50,000 crore annual export is given to the across the globe. We are the pioneer in exporting the drug. That's why India is called the world pharmacy. We are proud to say that we have as a regulator, as we don't act like a regulator, we are facilitated to the industries, we are facilitated to the academics, we are facilitated to the all the stakeholder which is coming to India. And you know, the conflict between India and China, WHO has categorically given a strong message to the Western countries, that is, if India is is not supporting in the field of formulation or in the bulk drug or in the medical, anytime they can capitalize the tactic policy for the creating artificial crisis to the healthcare vertical. So therefore, all the WHO member countries are facilitating India for technology participation, for academic participation, and for the all other aspect that is in the geopolitical situation of the world. today now in the government of India has taken proactive approach for giving subsidies and as well as technical know-how and as well as research incubation for the destination for India. We have almost we have at par. We are going to be the whatever the hurdles were there in a medical clinical research field, that we have a very stronger animal rule and utilizing human resource for the clinical trial. We have almost liberalized and as good as stringent norms, which is as good, which has been facilitated by US FDA, MHRA and TGA and Japanese regulator. So Japanese regulator is the most stringent regulator. They have acclaimed that India is going to be their partner in the field of medicine and healthcare. So far as all of you know, that is our almost 5,000 manufacturing units are having MHRA approval, US FDA approval, and TGA approval, EMA approval. So therefore, this world is looking towards India. And we as an Indian, we feel proud, all of you, to inviting for all your technological launchpad. You please do come to India. We are eagerly waiting for your as a facilitator, not as a regulator. I will facilitate. We'll promise you to give all the facilities, which is world-class facility, including the government subsidy, including the land subsidy, including the academic subsidy. We as a smaller state in the eastern part of the countries, we have almost manpower, almost 50 colleges, which is running pharmaceutical degree in the state of smaller 3.5 crore state of population of Jharkhand. We almost last 5 years, we have 54 colleges, which is giving you almost 6,000 per annum output for pharmacy graduates and diploma holders. So therefore, I will request that we have a 500 acres of land, which is going to for a specifically earmarked for the medical formulation unit, for medical devices unit, for bulk drugging unit. So in the Jharkhand, we are emerging with a new industrial pharma park policy. We are coming with a medical device policy, and we have invited as a single window clearance. You need not have to come to our state for any kind of thing. We will provide you for your doorstep services that I'm on behalf of government of Jharkhand, I'm promising you. I will share all the things which has been passed by the cabinet of the state of Jharkhand. I will share during the 2 days of the session. I will say every notification, every policy documents, as well as the expression of interest document, which has been legal, investor friendly, as well as academics friendly. We are also inviting medical colleges, almost 6 private medical colleges in the 1 or 2 years are coming to the state of Jharkhand. So therefore, I have a very hope for all of you. Please, as an ambassador of the state of Jharkhand, you please always make a positive impact that is in the state of Jharkhand. Although whatever is the past, we have a stigma on coming with a legacy of Bihar. Therefore, it was changed. We have changed the perception of eastern part of the country. That is, Jharkhand is emerging, is known for the metal and steel sector and minerals rich. We also want, that is, our people have this most economical, most reliable people, most skilled people in the state of Jharkhand. So government of Jharkhand is giving all training support. We have every field of the verticals in the pharma and allied industries, medical devices, food processing, that we are providing all the facility to the investor. And as a regulator, I am promising you, my name is Sumant Kumar Tiwari. I am remaining another 15 years for your doorstep services. Please do come to Jharkhand, and I welcome all of you for academics, for any kind of support. My 24-hour door is open for your any call, any suggestion. I humbly thank you, all of you. And I thank you, the organizer of Arka Jain Institute, for the Dr. Raji, our guru, is the Vice Chancellor, and Dr. Jyotirmay Shahuji for inviting me, all of this eminent guests. And thank you so much to all the students. My good wishes for just get the benefit of, you will never get such a eminent speaker, eminent across the field of all vertical of pharmacy field. They will participating. Their knowledge is just a boom for you. You're emerge with a bang, big bang in the field of pharmaceutical. My all features, my all blessing with all students and all those things. Thank you so much. Thank you.
Thank you so much, sir. And we are really privileged to listen from you
Thank you.
in sharing your promises to extend the services of the Jharkhand Drugs Control Directorate towards Arka Jain University and its pharmacy department. Thank you so much, sir.
Thank you.
I would request Dr. Arvind Fowler to offer the token of appreciation to Mr. Sumant Kumar Tiwari, Assistant Director Drugs, virtually, and this is being offered.
Thank you so much.
Please accept it, sir.
Thank
And
you.
on the screen, we are having Dr. Fowler.
Thank you.
Thank you so much.
Thank you.
Now, the inauguration or the inaugural session would end up with a formal vote of thanks. And I do not know personally from whom should I start. But
The participant from all other countries, those who are from the other countries, they should given first privilege, as I being a host.
thank you so much, sir. You have made my way easy. Thank you so much. Yes, the participants across the globe, they should get the first thanks who have collaborated with their participation to make it a grand success. I thank all the participants, delegates across the globe to make this function really a memorable one. I owe gratitude and thanks to the dignitaries of the day, Chief Guest, Mr. Sumant Kumar Tiwari, the Guest of Honor, Dr. S.G. Deshpande, Director, Faculty of Pharmacy, St. Pedro College, Dr. Arvind Fowler, Dean, School of Pharmacy, Arka Jain University, Dr. Jyotirmay Sahu, Patron and Dean, San Pedro College, Professor Fatima May Tesoro, President, San Pedro College, the Philippines, Sister Ida T. Franchilo, Vice Chancellor, Arka Jain University, Professor Dr. S.S. Razi, and all the dignitaries personally who have been here with us today in the morning to witness the inaugural session. I owe gratitude and thanks to all the program coordinators, deans of Arka Jain University, the faculty members, the technical assistants, and the IT assistants, the press and media personnel, and one and all who have contributed in one or the other way to formalize it, materialize it, and make it a great success. This podium thanks all the participants, including the dignitaries and the delegates once again. And here before I hand it over to Ms. Khushbu Raj for the plenary session to be conducted ahead, I say goodbye and salamat po, the Philippines. Thank you.
A very good morning to all of the participants, dignitaries, delegates, and my dear colleagues. First of all, I would like to welcome you all to this international e-conference. This global meeting will assemble the world leaders in the expanding fields related to the various natural and novel approaches for pharmaceutical research area. So now moving to our plenary session, I would like to invite our first speaker, Dr. Krishnapriya Mohanraj, Professor of Pharmaceutical Analysis, Bombay College of Pharmacy. We all welcome you, ma'am, and we are very delighted to have you for our today's session. So talking about ma'am, she has done her M.Pharm PhD from University Department of Chemical Technology, Mumbai. She is an educator and researcher for more than 25 years. She holds 26 publications, 23 presentations, 1 patent, and 1 technology transfer in her credits. Now, I would like to request to Krishnapriya ma'am to kindly share her valuable words with us. Over to you, ma'am.
Thank you. Am I audible?
Yes, ma'am, yes.
So I will share my presentation. First of all, good morning, and very, very happy to be part as a keynote speaker at this e-conference. So I'll share screen. It's coming. Host is disabled. Participant shares screen sharing. So can somebody enable it?
Yes, ma'am, you can share the screen.
Is it visible?
Yes,
Is
ma'am.
my screen visible?
Yes, ma'am, it is visible.
Okay. So first of all, I would like to thank the organizers, Arka Jain University and San Pedro College, for inviting me to be a keynote speaker at the e-conference on natural and novel pharmaceutical research. So in this, I would like to mention here that when we talk about pharmaceutical research, and the research that I have done, I'm talking about a researcher's insight. Okay, so the researcher being me and my team. So a researcher's insight into natural novel pharmaceutical research. So we'll provide you a little insight on to the world of research that we and my research group and what I did during my PhD days at ICT. So, and my research at Bombay College of Pharmacy. So when we talk about natural, okay, I've done a lot of work which is related with natural. And there are some things which are not directly related to natural, but they're still novel. And I would say that all natural researchers also, any research that we do, we do novel research. It has to have some novelty or newness in it. So the first, what I would be talking about and taking up as a case study would be bioactivity guided fractionation of herbal extracts. This is some research arena which is very, very close to my heart, because this is what I did during my PhD at the Institute of Chemical Technology under the guidance of Professor K.G. Akamanchi. So this neem leaf extract we had explored for antiplatelet activity. And now, as a researcher too, my group is working on Jaifil or Myristica fragrance for its antifungal activity. And here I would be taking a case study just to show you how hyphenated techniques like LC-MS-MS can help in finding out what are the components present in a particular fraction. Next, we'll go on to the CAM assay. So many a times what happens is that when we do research, we may not only have to find out something unique in the natural product, but for what we are searching for, but we may have to develop techniques which will help us, do our research better. And especially here, I would like to mention with respect to the CAM assay, this is for anti-angiogenic activity, choroallantoic membrane of the chick embryo, in which has a lot of blood vessels, and when we have anti-angiogenic activity is good for cancer and a lot of other conditions like macular degeneration. So when we are doing this particular assay and seeing something visible, and it may be totally objective, how can we develop something like a scoring pattern which will help us convert something which is qualitative into a quantitative? So in this, we'll be looking at hemidesmus indicus, anti-angiogenic activity. And I must say the initial work on this, which I've not been mentioning, means covering today on VEGFR inhibitors of chalcone analogs. This was a DBT, Department of Biotechnology grant, which we received, and Dr. Dipti Gatne was my student, and now she's a faculty here at Bombay College of Pharmacy, and she's the co-guide for the research work that we have done with hemidesmus indicus. Next is when we talk about novelty, sometimes our research itself will give us ideas for further research. And this was when my MPharm student Jaysha, he was working on benzotriazole analogs, okay, for antifungal activity. And we had done it by 3 methods. Benzotriazoles were supposed to act on lanesterol 14 alpha demethylase. Okay, that was the literature report of the action of benzotriazoles. And so we did docking studies. Since lanesterol 14 alpha demethylase is involved in ergosterol formation, and if ergosterol is the biosynthesis is hampered, then you're going to have fungicidal activity. So we did even a sterol quantification assay by UV, and of course we did in vitro antifungal activity, but there was no good correlation between all the 3. And, you don't just leave it like that. Our hypothesis was that maybe these agents are not acting only on lanesterol 14 alpha demethylase, but maybe acting on more fungal targets. So if they are going to act on more fungal targets, they would be useful in, acting on resistant species like fluconazole resistant species. Okay, then we proceed to doing chiral chromatography. Again, here you say sometimes, that there are circumstances that make you lead to do some research. Like the circumstance here is with respect to us applying for a grant to the University Grants Commission. That was the year when Bombay College of Pharmacy planned to start, was starting the M-Pharm pharmaceutical analysis, and I was heading the department. So, and we had an HPLC, which had a UV detector. And we thought that, why not get a PDA detector, okay, photodiode array detector. And in that process, we wrote, a project called multi-component analysis of drugs. So when you have multi-components, then, you find that a PDA detector is useful. I thought since we'll have so many M-Pharm students, so we can give something like, fixed dose combination stability indicating method to a student. Then another student can work on herbal analysis, because herbal analysis definitely has so many components, and that's a challenge that you will find when you work with natural products, multiple components. So herbal analysis. Then other area is again a form of bioanalysis, that is and therapeutic drug monitoring, where you will find that along with your drug, you have the matrix components, and you have to separate them or do metabolite analysis. So all this definitely are multi-component analysis. And we put in chiral chromatography because we had experience of working on enzymatic resolution of drugs. And, for that also, we required chiral chromatography. So, and we had experience in that particular arena. So we asked, but that time when we were doing the enzymatic resolution, we had only, a normal phase chiral column, and the solvents used in that very, very expensive. Okay, N-hexane, isopropanol, ethanol, HPLC grade. And what was happening was by the time we had applied for this particular grant, there was, newer immobilized columns where you could use, polar organic solvents also. Okay, so that is the immobilized chiral columns. Thought, let's write for PDA detector and for chiral columns. And the agency gave us PDA detector, but did not give us chiral column. And of course, in the project, one part was chiral chromatography. So that becomes, invention sometimes comes through necessity. So we thought of why we cannot use some naturally acting compounds, natural products. See, many of the natural products are chiral. Okay, so we can use, chiral mobile phase additive and try and do, chiral chromatography without using chiral columns. And we'll show you one part of this. We also are very lucky at Bombay College of Pharmacy for a project that we wrote to the Department of Science and Technology under the Drugs and Pharmaceuticals Research Program. They gave us, funding of 5.87 crores for setting up a national facility for research and training in integrated analytical strategies for discovery, development, testing of drugs, pharmaceuticals, and nutraceuticals. So we do a lot of research in this, as well as, impurity profiling using LCMS-MS, HPT-LCMS, ICP-MS, a lot of it. It's not possible to cover all these things. I know I made a very big presentation. I had to cut it short when I practiced with, for the time that is remaining. So with this background, I would take you now to a few case studies, and we'll go a little fast because we are running little short of time. So, but the important parts we'll definitely cover. So when we talk about drug discovery from natural sources, I will just take all these, you have 5 points that very, very important that we have to cover. And by experience, one is your source of your material, and always authenticate your sample. You might take it from a very, very reliable source, but don't forget to authenticate. In the case of Hemidesmus indicus, we found that, when we sent it for authentication, they said, no, this is not Hemidesmus indicus. Then we repeated authentication with another agency, and still they also confirmed it was not. So very, very important to have authentication. Keep a sample in the herbarium of what you have used. There are seasonal and geographical factors. Like when I started work on, natural products under at ICT, and I wanted to work on antiplatelet activity, and I thought, we tend to use logic so many times. So aspirin is, of course, having anti-inflammatory properties, and it also has antiplatelet activity. Mechanisms quite similar. Okay, so the COX inhibition and cyclooxygenase and thromboxane A2 come in the prostaglandin biosynthesis pathway. So we thought that let's look at something herbal. And as Deshpande sir, who is my teacher, mentioned in his chief guest address, that, Ayurveda is so strong in India. Then I thought that Dashamula Arishta, with 10 roots, we can, you will find a lot of literature on, the various activities of it, but no antiplatelet activity reported. So we thought that let's look at these, 10 roots. And I remember Dr. Vadlamadi Rao, who was my teacher of MPharm pharmacology at ICT, and who was later working at Hex Research Center, whom I approached, and they also did a lot of work on antiplatelet activity, advised me that if you look at Dashamula, the North India, what are the 10 roots? And in South India, what are the different 10 roots is different. And he told you want to do a PhD. Even if you take one plant and you try to do bioactivity guided fractionation and try to isolate it, take you so long. So there are these seasonal geographical factors, extraction methods, whether you do a decoction, oxalate extraction, maceration, it will all make difference. The nature of extracts are usually hygroscopic. Half the thing that you are weighing will be water. So you have to see that your samples are dried very well before you do the activity testing. And as I said, there are millions of constituents present, and the active constituent may be present in very, very less amount, and then to isolate that becomes a challenge. This is an article from 2020 Journal of Natural Products, where you can see that the synthetic compounds, purely synthetic, is just a cotton. So you have the biologicals, the natural products, botanical drugs, that is mixtures, and you have your vaccines, and you have the synthetic drugs which have come from a natural pharmacophore. Okay, so and if you look, this is data up to September 2019. You can see that how the biologicals have increased, and the natural products, only this S is synthetic. All the others are synthetic, mimetic, or having natural product mimetics. So you find that there is definitely a lot of things with respect to the emphasis in the current drug pharmaceutical world, emphasizing on biologicals, vaccines, and of course on natural products. Okay, so this was my neem leaf extract project. So first case study, I've already given you a background, thankful to my guide, Professor K.G. Akmanchi. And we did this in collaboration with the GS Medical College, where we had 3 doctors, Professor Dhanukar, Dr. Urmila Thatti, and Dr. Nirmala Regi helping in this project. I'll go fast now in the case studies. So when you have to do drug discovery, you have to identify a disease or the therapeutic target, develop the methodology, and then you can either do rational drug design or selective screening. Screenings can be with synthetic or with natural products. With natural products, better to do bioactivity guided fractionation. So you have your lead compound, then identify it, and then do modifications for lead optimization. Okay, so for the neem leaf extract, we did chloroform extract, soxalate extraction. So this was got from Dabur. I had 1 kg of material got, and you did soxalate extraction, removed all the, the fatty material with chloroform, whatever residue was there, treated with methanol, because what is it that when we were testing, we used to test it on platelet rich plasma using a platelet aggregometer in which the drug would be added. So we could use very less of, very less quantity of any organic solvent in it. And if I use very, very non-polar compounds, it would lead to, precipitation in our plasma. And so we stressed on using the methanol soluble extract, which is found to be active, insoluble, inactive. And then we have done fractionation using water and butanol at 2-point fractionation, and the butanol extract showed activity, which we used Cefadex LH20 and got many fractions. LCMS revealed the presence of flavonoids in this. And then we proceeded to do semi-prep HPLC. And this was how we isolated rutin. Sometimes it's serendipity. Okay. So we didn't have a semi-prep HPLC at ICT. And so I'd gone to the company Searle. And the operator asked me, okay, we had this fraction, which was there from what I had got from Cefadex LH20 running, told that please, we added the mobile phase, and she asked me to go and sonicate it. And when I went and sonicated it, you wouldn't believe they got an orange precipitate. Okay. And then when we centrifuge that, and we could isolate a single component out of it. So sometimes you get lucky that you have trying to separate through such a complicated instrument, and finally you get it all with a simple technique. So that's research. And later we found that even after rutin was separated, the supernatant was very, very active, much more active than rutin, much more active than aspirin. And, we had fractionated with high-speed counter group. I'll show you the results, and then went for LCUV. So this is how, I got an authentic sample of rutin, compared it by mixed TLC with my centrifuge sample. It was the same. Of course, the washings and supernatant also had a little amount of rutin remaining. But if you see the results, you can see percent inhibition. Okay. So that's the orange one is percent inhibition. The blue is the maximum percent aggregation. So you find that the percent aggregation in the vehicle control is the maximum, but the inhibition is nothing. And you will find that the supernatant had very good aggregation and inhibition of activity, as also aspirin and rutin and whatever we had separate called the centrifuge sample. This is the structure of rutin. We analyzed it using the NMR, and you can see that whatever was left, we did high-speed countercurrent chromatography of the supernatant, and you can see 82% inhibition, okay, of platelet aggregation. But more clear in this chart, much better than what is aspirin or this is for all fractions, and my centrifuge sample, we use 100 microgram per ml and aspirin 50 microgram per ml. That's why the previously you can see they are, when they are in the same dose, they are quite similar activity. But then later when you increase the dose, the activity is increased. And this is fraction 12. And, by the time they pushed, we've been pushed out of come finish your PhD work. And we did an LC-UV. This is how the platelet aggregometer scans look like. So you can see this is for my fraction 12. This is for aspirin. Every time you compare with methanol, these are other fractions. Okay. So the fraction 12 had 4 components. And later when I even started doing my research, this work is patented. And it's Professor Akmanchi's first patent, from his students. So I'm very proud of this work. And we later wrote for, many funding agencies to continue this work, but didn't happen. So we now go to the next. This is Jaifal, what is called as nutmeg. And in this, I'll go fast. Many fractions which are active. Okay, these are the active fractions, and these active fractions later they were LC-MS analysis. So that's what from this particular study I want to show. Okay, we were doing all our antifungal activity using RAMA, Resazurin microtiter assay. The blue shows no growth and the red shows growth. So, this is sterile water, doesn't show any growth. This is fluconazole in very, from high concentration, it gets diluted. So you're diluting it, half, double dilution as it is done. So this becomes your MIC, where there is a difference between, there's no growth and here growth. So your MIC is between this. So MIC of fluconazole is very less, and for the Jaifal extract and fraction 1, it's comparatively higher. But as you go and hexane extract, as you fractionate, you get better activities. Okay. So it was also found that sometimes you may get the chloroform extract was giving us very good activity, but when we fractionated, then the activity comes less, maybe because it must be acting synergistically. So that is one more thing that you have to take care while working with natural products. Okay. So identification is done by through two LC-MS techniques. One is a Shimadzu triple quadruple instrument, and other is ABHISHEK Q-trap. And just comparing some, see, in LC-MS, whenever you are doing, it's very important that the sample ionizes. So you can find here that fraction 1, with the tuning parameters which were used for the Shimadzu instrument, gave myristicin present, but nothing in the ABHISHEK's Q trap because it didn't have, the ionization didn't take place. Fraction 3, Shimadzu, methoxylicarin, I'm going fast. Even ABHISHEK's 5-methoxylicarin B with fraction 5 in Shimadzu myristic acid, and in fraction 5 with SciX, it is licarin A. With chloroform extract fraction 8, it is isoelmicin, elmicin, and methoxyeugenol from Shimadzu experiment. And with the SciX one, we have isoelmicin, elmicin, or licarin C. This is another work which we have recently done for determination of LC-MS analysis of flavonoids in Dracobel TM new Dracocephalum moldivica aerial part extract. So you can see different flavonoids and flavonoid glycosides which were identified by LC-MS analysis. And the same thing, we even did an HPTLC analysis, and you can see that with various standards of flavonoids and what is present. And here I would specifically like you to notice here, this is test sample ethanolic extract, test sample chloroform extract. As you change the whatever you use for extractions, what you're going to get components is going to vary. Then we hydrolyzed this, the chloroform extract, and then did so, the glycosides, they, hydrolyze, and you see more of the non-polar components in it. Another thing, again related with natural product, is gum acacia. And in gum acacia, you will find that 3 pharmacopoeias give different methods. And then we did a comparison of all those methods. And then we found that definitely the JP methods, this is glucose, which you can see here. These are different sugars. Okay. And the second one is glucose. The test sample, the gum acacia doesn't have glucose. It's not supposed to have. And this by Japanese pharmacopoeia, you can see there is no pink spot. This is very, very clearly visible. And this is by European pharmacopoeia, not much resolution between galactose and glucose. Even if it is a mixture present, it's very lightly seen. So, when you compare the methods, it's the Japanese pharmacopoeia which works. Okay. So next we go to the CAM assay development. So I'm not going to speak about, bioactivity guided fractionation too, or this thing, only that how different extracts you will get, and how for something which is very, very qualitative, we have developed a scoring pattern. Okay. So it is Ananthabol, Hemidesmus indicus, only one report of a decoction having anti-angiogenic, but not through CAM assay. And we thought we would try the other extracts too. So there was decoction, maceration with methanol, maceration, hydroalcoholic ethanol water. We even did methanol soxylate extraction, chloroform extract, and chloroform and hexane also soxylate extraction. The yields vary different, the maximum yield being from maceration 70.26%. And then when we looked at, before we did the CAM assays, we had to decrease the dose. So all these we did HPTLC analysis, and we had, it is lupiol is reported to have anti-angiogenic activity. Lupiol is present in Ananthabol. So we just, did a study and saw with vanillin sulfuric acid reagent. And yes, lupiol is present in some proportion. And before we went to the main CAM study, we had to do toxicity study of the doses. find the dose range finding study, and then do the pilot and the main CAM study. So the CAM assay is you use chicken embryos, day 0 procurement, day 4, a small hole is made. Day 0 is that when it is hatched. Okay, that is day 0. By day 4, you have to start. You can't take eggs which are quite old. And you make a small hole on the top with the 18 gauge on the air sac side and inoculate with 10 microliter of test vehicle control and seal with parafilm. Then on day 12, these are kept for incubation. Day 12, harvested and fixed in 10% formalin PBS solution. And then there is H&E staining and then stored. So here, this is how a CAM looks like. Okay. And for doing toxicity studies for all extracts, okay, 0.1 to 1000 microgram per ml was checked. And also for the positive control from 0.1 to 1000. And what was found was that decoction, maximum dose, that is no death occurring, or, it's with respect to the eggs, we could go up to 1000 microgram per ml. But for these two, that is methanolic macerate and methanolic soxalate, 100 microgram, chloroform and hydroalcohol, 10 microgram, and docetaxel, which is it's an anti-cancer drug and anti-angiogenic. It gives 1 microgram per ml. Okay. And now I'm just going to show that how we developed a scoring pattern. Okay, scoring pattern with the, what do you say, the CAM assay. So this is how a CAM looks. You will see that it has a lambda shape. Here, can you see this lambda? Central vessel. So there are 3 things that we are going to look at. Central vessel, the branching pattern, okay, and collateral vessels. Collateral vessels develop from the bottom. Okay, so you will find that those do not have a central thing or something like that. And as we progress, we will see that the central vessel becomes smaller, smaller, smaller. The branching, normal tree branch changes, you get parallel vessels and you will get bendings, crossovers. These are all simple, straight, and then each thing branches. But there's no crossover of any vessels. Okay. So, and the collateral vessel is very, very less and minimum branching of this. But as you see anti-angiogenic activity, the whole pattern will change. So this is rank 0, then rank plus. So in this, you can see it is sustained, there is slight disorganization, some crossover, okay. Then +2, you can see distinct sharp turns in vessels. Yes, sharp turns in vessels. Okay. Then you see, again in the same +2, you will see major crossovers. So you can see that one vessel is crossing over another vessel. See, so many like this. So this is all +2. And in the collateral thing, there is collateral vessel development, which can be seen. Okay. And next, +3, what you see is long parallel vessels. Okay, not like a tree branch, but it's just like parallel vessels. And major collateral vessel development, you can see from the bottom. And we come to rank 4. Okay, this is the highest. So you can see the central vessel is totally distorted. Either it is short or it is missing. The length is reduced. Multiple vessels from the upper side of the membrane. So you can see it when you open it. Multiple. Otherwise, you have just one central vessel. Okay. Then these are all wavy, lot of disruptions, and even in collateral, extreme disorganization and distortions. So this is +4. So we'll show you, this is how a real cam looks in a vehicle control group. And when it is rank 4, just see the difference. I'm going back. See how this is how a regular cam in vehicle control looks. And this is with +4. And that's also remarkable reduction in size. So this now scoring pattern, how do you put it into a quantifying thing is, okay, this is how the scoring pattern thing for the various extracts came. And you have to quantify it. So the number of eggs with score 1 into 1 you will do, number of eggs with score 2 into 2, number of eggs with score 3 number into 3, and number of eggs with score 4 into 4, divide by the total of number of eggs. And then we made this pattern of no anti-angiogenic, weak, good, and good anti-angiogenic activity. And we find that positive control are very good. The others see 2.8, between 2 and 3. The rest, this is above 3 and this is between 2 and 3, good anti-angiogenic activity. We also tried different other parameters like area of cam, length of cam. You can see this over here. Vessel density, width, area, branching points, which were all done. And vessel density was a better system. I'm not going into details of this. Okay, this is the the first project that we did on anti-angiogenesis, Department of Biotechnology. This was chalcone analogs we got it from natural products. There are many chalcone analogs which have anti-angiogenesis activity, and we developed, synthesized more and did this activity, but I'm not presenting this work. And we'll now go to case study 5, where in M-Pharm, I told that there was no correlation between the in silico docking study, ergostrol, and in vitro, and the hypothesis was, let us look at, 27 fungal targets and few human cholesterol, cholesterol synthesis pathway. These, when we have checked, we have even found that fluconazole, okay, which we thought that also is acting only on lanestrol 14, is acting on 8 out of that 2 are from human, okay. So whatever we wanted at least 7 targets it should act on. So inhibit 7 or more. This predicted inhibition is not possible to cover everything here, but we have done 2D QSAR, we have done pharmacophore modeling, we have even done homology modeling to make the enzymes which are fluconazole resistant lanesterol 14 alpha demethylase based upon literature reports of the amino acid sequence. And finally, we shortlisted 25 compounds for synthesis, out of which 7 we use, which we know will not have activity. So that, it shows that our model is good, that, when you plan and design something, this is going to have very good activity and this is going to have less activity. Finally, it should show those results. And we have used in vitro analysis, and you can see this is, sorry, I don't know why this is coming on top. So we have fluconazole resistant, the MICs and MFCs were found. And you can see 27, 30, 69, 78, these all showed very, very good activity. And you can see over here that against all of these compounds, from 1 to whatever compounds, 25 different compounds that we have synthesized, from 10 microgram to 200 microgram per ml, we've done the active against fluconazole susceptible fungi. Okay, lower concentration, no, but higher concentration, some activity is there. But when you look at fluconazole resistant C albicans, it's only these 4 compounds that we designed, and itraconazole C. Fluconazole also does not have activity. Okay, so then we went and did this for in vivo study. So this is an in vivo study in murine candidiasis model. And you can see that when we've done the study, itraconazole, we got permission from ethics committee only to test 2 of them, 2 of the compounds. So in 10 milligram per kg, 20 minutes. I want to tell here that when we did toxicity, these are all more than 2 grams per kg. That means not toxic as per OECD guidelines, while itraconazole definitely is only up to 500 milligram per kg weight we can go. Okay, otherwise it is toxic. So these compounds are not toxic. You can see that they are very much concentrated in the kidney, the followed by the brain, lung, and spleen. And definitely there is very, very, very good activity from the colony forming units. Okay, coming to the last case study, I just want to ask whether I have time to cover this. Hello? Hello?
Yes, ma'am. Yes, ma'am.
Okay, so I'll do fast. We started a little late, no? So we have, when we talk about this one area which we work, so go fast, chiral separations. So we have, they are similar in everything. Enantiomers is similar in everything, only they rotate plane polarized light differently. And they have a different response in chiral environment. So only when you have something chiral can you resolve chiral. So, and why should we separate those enantiomers? They have different biological and therapeutic effects, different pharmacokinetic, and all of you are familiar with the thalidomide disaster in 1960 and US FDA guidelines in 90. Here, you had fecomalia, the babies, the mothers took thalidomide for morning sickness and babies were born with seal limbs. So US FDA guidelines in 1992 tells that if a compound can be marketed as chiral, it has to be marketed as chiral. And if you want to market it as racemic, you have to give a valid reason for that. Okay. And the innovator companies always do that. When the patent is ending, they will go to a chiral switch. Okay. So how do we do chiral separations? Is you have enantiomers. So you have the S and the R. So S and R cannot be resolved by any means unless you convert the enantiomers into diastereomers, even if it's a transient diastereomeric complex. So you take something else which has a chiral environment but pure. So you will have SR and RR, okay, forming the diastereomeric complex, and then they will get resolved. So chiral HPLC of direct technique using chiral stationary phase or chiral mobile phase, or you can do a chiral derivatization and then use a normal column. So now we're going to concentrate on chiral mobile phase additive. Again, from natural origin, cyclodextrins are there. There are a lot of proteins, macrocyclic antibiotics, and ligand exchangers used as chiral mobile phase additives. We started with cyclodextrins because they are easily available and they do not have UV absorbance, stable over a wide range, and inexpensive, especially beta-cyclodextrin hydroxypropyl sulfated is expensive, but we synthesized that so that the cost becomes less. So this is beta-cyclodextrin, how it looks. And you can see here how that moiety, the drug, chiral drug, how it's going to get separated is here. We also used a technique of core shell silica columns. So the core shell is the inner core it's not porous, and only the outer shell is porous. So suppose we are using a fully porous, the compound has to come right into the center and then leave out. While here it leaves out in less. So what happens is the retention times are lower, the total analysis time becomes less, and that very, very sharp peak. So you can see from one only, we have done 12 compounds which we have resolved. All compounds do not get resolved by using sulfated beta cyclodextrin, but we've done 12 compounds, and I'm just going to present and show you one particular one, the milnacipran, which we worked on first. Okay, so this is the HPLC. This is my first AICTE project. The first government grant that I received was bought in 2002, and I'll say it is still working, my JASCO 2000. And this is from this new grant of DST DPRP, where we have a UHPLC as well as an HPLC system. And I told you we've tried beta cyclodextrin, hydroxypropyl, and sulfated. If you see the cost, 5 grams sigma aldrich cost 7,000 and more, and cyclolab 5,000 and almost 6,000. So we synthesized sulfated beta cyclodextrin using sulfamic acid, and the cost of it is just around 500 rupees. Okay. And we have done the technology transfer, which was mentioned, to Loba Chemists for them to commercialize it, but still they have not done it. So I have told that in 2 years, if you do not commercialize, then we can take it to some other company. Okay, HPLC method development. For milnacipran, it's antidepressant drug, and the levomilnacipran is more effective, and the US FDA has told that, it's better to give the levomilnacipran, and we've done the formulation which we got from USA. Okay, so just in brief to give you an impression. So this is a HiQ cell column which we had before, and on that we did, and then we had kinetics column, and see the difference. But then we thought this is 250 millimeter, my kinetics is 150 millimeters. So then we purchased a hyperclone C8 of 150 and compared. Is it due to the length? So it's not due to the length that you are having less. And see, less than 4 minutes for the analysis with a resolution of 2.96. So you can see 2.96, we optimize the sulfated beta cyclodextrin concentration, the pH, and the mobile phase composition. And what was finally done was 10 millimole sulfated pH 3 using acetonitride, and kinetic C8, which is the core shell column. You can see the number of theoretical plates. We should have minimum 2,000. The number of theoretical plates are 8,000 and 9,000, and the time is within 5 minutes the analysis is done, and we validated the method, and we have seen for Fexima capsules from Forest Pharmaceuticals, seed, levomilnacipran, a single enantiomer, there is no dextro at all. Okay, even molecular docking studies were done, and it was seen that sulfated, there is more difference, and maybe that's why only sulfated worked and hydroxypropyl and this did not work. I acknowledge my guide, Professor K.G. Akmanchi, and my alma mater, Institute of Chemical Technology, Bombay College of Pharmacy, where I work, the DST, DPRP for the sanction of grants, AICTE, and Amrit Modi Research Foundation for, Jay's work, benzotriazole work, DBT for Dipti Gatne's work, and my students all of them who have contributed for this particular, which I could present. I'm there because of my students. Mr. Avinash Barsha, my new PhD student, he's under the Prime Minister's doctoral fellowship scheme, which he recently got. And, thank Arka Jain and the Pharmacy Department of San Pedro College and all participants. Thank you very much.
Thank you, ma'am, for this wonderful session. It was very informative for us. Now, anyone have any question? Please ask. Yes.
Thank you so much, ma'am, for this nice presentation and beautiful work. And I really appreciate how you presented. Ma'am, I have certain questions since I am not from pharmaceutical background. I just want to ask you whether the genes involving these products or bioactive compounds, all the genes or the metabolic pathway genes, if we are able to clone all these genes, so because it contains enormous number of genes, if we are able to clone even some of them, will it be helpful at some level, clone and express them in bacterial system?
Yes, that would definitely help. And I remember when I was working in ICT, the foods department, food and fermentation technology department there. So, no, sandalwood, you get the sandal, the perfume. So from, doing tissue culture, okay, not even going up to gene level, just by doing tissue culture in a manner such that you increase the production of the constituent that you need. Okay, so that is one thing. Plus, I will tell you, like when I worked on immobilized enzymes, okay, my enzymatic resolution worked on immobilized enzymes. So it was from what you get, the immobilized enzyme is not from the organism 5H, okay, Cal-B, okay, the lipase Cal-B was not got from Cal-B, but was got from bacteria, okay, which were engineered, genetically engineered to produce this lipase. So, it's not only in the pharmacy field, but definitely gene and gene-based, what do you say, the interventions can definitely help in increasing one particular kern. You can do genetic engineer, or how is your biopharmaceuticals made? Your biopharmaceuticals are made, by genetic engineering, so by making your fermentation technology such that you would get your material. Okay.
Ma'am, still I'm a little bit confused regarding that if we are not able to generate that bioactive antifungal compound, but if we are able to generate single metabolite, means the metabolic pathway, if I am getting single product, but I am not able to express all of them.
No, you can do screening of it. You can do screening of it.
Okay.
And see whether it is active. You can screen against multiple targets and multiple activities. Okay. So that's how, work was done at Hext Research Center. People who used to work with natural products, anybody goes to their native place, tells you bring soil from your native place, then there'll be some different microorganisms there, and then that fermentation was done, and much more different metabolites used to be there, secondary metabolites, and then they used to screen that.
All right, ma'am. Okay. So अगर मैम अगर पूरे प्रोसेस पूरे पाथवे में से मैं सिर्फ एक ही कंपोनेंट आइसोलेट कर सकती हूँ और एक्सप्रेस करा पाती हूँ बैक्टीरियल सिस्टम में, that is also useful.
It's useful, but it will be active. Nobody can guarantee. You will have to screen and see. So you put that, then you will have to check for what its utility will be.
I need to check it what its utility will be. Okay, so
Yeah.
what platform can I use to check the utility of that particular derivative?
No, you will have to check the different platform. No, suppose I'm doing antifungal, then antifungal testing karna padega. If I'm doing antiplatelet, then I have to test antiplatelet. If I'm doing anti-angiogenesis, I will have to check for that. So it's all different platforms.
All right. Thank you so much, ma'am.
Welcome.
Thank you, ma'am. Now I would like to request Dr. Erwin Feller to kindly felicitate Krishnapriya, ma'am. Hello, sir. Please switch on the video.
Thank
This
you.
is a small token of appreciation, ma'am.
Thank you. Thank you very much.
Presented by Dr. Erwin Feller on behalf of School of Pharmacy, Arkajan University.
Thank you.
Thank you, ma'am. Now moving ahead for our next session. Today we have Dr. Mona Simalti, co-coordinator of Swam Sale, Assistant Professor, Department of Pharmaceutical Science, HNB Garhwal University, Srinagar, Uttarakhand. Ma'am has 18 years of teaching experience with 31 international and 37 national papers in various reputed journals, 4 books and 2 Indian patents in her credits. We welcome you, ma'am. She is one of the most cited researchers with high index 24 with citation more than 1700 and total impact factor more than 80. We welcome you, ma'am. And now I request you to kindly share your knowledge with us. Over to you, ma'am. Ma'am, you are not audible. Please unmute yourself.
Okay. Thank you. Thank you for introducing me, and I'm quickly starting my presentation because we are already ahead of time. I think we are late 15 minutes, around 15 to 20 minutes late. Okay. So can you see my screen?
Not yet, ma'am.
Now?
No, ma'am.
Just a minute.
Yes, ma'am.
Yeah, can you see now?
No, ma'am.
Okay, I'll stop sharing and I'll again reshare.
Ma'am, I guess it is a network issue. Ma'am, can you send us your presentation so we can share here? Hello. There's some technical issue. Ma'am will join within 5 or 10 minutes. Stay with us. Thank you for your patience.
Yeah, am I audible now?
Yes, ma'am, you are audible.
Yeah, I'm so sorry. Actually, I lost my network and actually I'm using Dr. Simulti's laptop. So anyways,
It's okay,
just,
ma'am.
yeah. Okay, now my slides are visible.
Yes, ma'am. It is
Yeah.
visible now.
Yeah. Okay, all right. Okay. So welcome dear participants in this session. So today in this session, I will be discussing about hair growth promotion and some hair growth promotion. What are the remedies? What are some herbal and synthetic drugs used for hair growth, as well as animal model which are used for carrying out in vivo study, as well as in vitro hair cell culture. About I'll discuss this topic today. So very quickly moving to introduction. See, dear participants, as we all know that hair are very important sociologically, and hair disorder, especially when it is severe, is profoundly affects the life of those afflicted. So alopecia, it has been recognized for more than 2000 years. So all we all know that it's psychosocial problem for those also who are suffering alopecia. So I'm skipping just few slides as and I'm directly coming to the main portion of this presentation about hairs. Okay, so hair, this is also, we all are familiar with hair is a protein filament that grow follicles found in the dermis. Okay, so these are simple thread-like structure about 50 to 100 micrometer in diameter. So two types of hairs, vellus hairs and terminal hairs. We have vellus hair, short and fine hair present on body, mostly on in infants, and terminal hairs on of head, eyebrows, eyelashes. Okay. So composition of hair basically consists of hair filament, which is primarily it is composed of alpha keratin protein, and hair follicle, hair shaft. These are important. Hair follicle, which is beneath the skin, and hair shaft, hair filament extended above the skin. Hair follicle, which compromises the following: hair bulb, hair papilla, hair erector muscles, sebaceous gland, and hair shaft. Okay, so this is basically the anatomy of hair. I'm just giving you a little insight of it about hair shaft. So we need to understand it because what we need to know in today's session is about the different model which we will be discussing. So when we will apply or will adopt some animal model for research, okay, research, because this is one of the very crucial topic for research and having a lot of scope in it. scope as in for PG dissertation as well as for PhD. For anyone who want and looking for a burning topic in the field. Okay, so first discussing hair filament, extending hair filament, basically extending above the skin and composed of three distinct layers: outer layer, that is cuticle, middle layer, cortex, and inner layer, medulla. Okay, so cuticle, cortex, and medulla. Just look at the slide, composed of 6 to 11 layers. Cuticle cortex composed of tiny keratin fiber which run parallel with each other along the whole length of hair follicle, and medulla, that is inner region of hair shaft. And this is hair structure. I'm quickly giving you the hair structure, the three main important parts which you can see: cortex, medulla, and cuticle. Okay, generation of hair. See, generation of hair, which is a very complex process basically, and it involves several processes such as hair differentiation, epithelial, that is mesenchymal interaction, stem cell augmentation, pattern formation, apoptosis, and cell and organ growth cycle, and pigmentation. Okay. So generation of hair, germinating hair, that is which are in the stratum germinative layer of the epidermis, which is the outermost layer of the skin. It penetrates downward into the dermis to form the outer boundary or lining of hair follicle. Then the layer below the epidermis, which is called dermis, and it grows towards the upward in upward direction into the follicle base. The enlargement of bottom part starts to form a massive actively growing cell called as hair bulb. Okay. So moving to next slide, generation of hair, that is follicle formation. So germinating layer of epidermis, then it merges with the root sheet which forms inner wall of hair follicle at the base of hair bulb. Basically, this is this is a mechanism of generation of hair, and once epidermis, which then merges with the root sheet and it forms the inner wall of hair follicle at the base of hair bulb, then formation of germinal matrix starts from the outer root sheet. Then the germinal matrix then grows to form inner root sheet. Okay, so hair follicle, which basically undergoes into 4 cycles. So this is important part which you need to understand little, that hair follicle cycle, which consists of 4 phases. Basically, the formation of hair follicle, it undergoes in a cycling process and having anagen, catagen, telogen, and exogen. These are 4 phases. So anagen, we consider it as growth phase, catagen, degenerative phase, telogen, resting phase, and exogen, that is shading phase. All right. So alopecia, which is which we are going to discuss. Alopecia, which occurs when anagen phase gets prematurely terminated and catagen phase, catagen's cycle starts too early. Okay, when it happens, there is alopecia, right? So telogen to anagen, then catagen phase, and then catagen to telogen. So as you can see in the diagram, that telogen 3 months lengthy phase it is, and hairs are shed at the end of this phase, and hair follicles enter into the new cycle after the end of this phase. This is telogen. Resting phase it is, anagen, which is growth phase. So epidermal cell divides to produce precursor cell, and the differentiation of the precursor cells to form the hair cell, and formation of new hair filament, 2 to 8 years lengthy process it is. Okay, and most hair remains in this phase. Okay, which phase? Growth phase. Okay, so this is about the hair growth cycle, hair follicle cycle, which consists of 4 phases. And now I am moving to alopecia, which is basically what alopecia is. Alopecia is hair loss, which is a natural daily phenomenon, and every strand of human hair on human head is genetically programmed to undergo growth, stabilization, aging, and shading. So every day human head sheds around 50 to 125 years, depending on the sex, whether male or female. Okay, which are replaced by regrowth of hair filament. Okay, and then our hairs are in in a cycling phase, or 4 phases, which we discussed in the previous slide. Okay, so problem starts when the rate of hair shedding or hair loss exceeds hair regrowth. Okay, so there the problem starts, or the regrowth is weak and unhealthy. So loss of 100 hair per day is considered as a normal, but if we lose more than 100 hairs per day, it constitutes a pathological effluvium which may lead to alopecia. Okay, baldness, it is also called as baldness, and which is an inflammatory autoimmune dermatological disorder, and medically classified into androgenic and androgenic alopecia, alopecia areata, and alopecia totalis, and last one, alopecia universalis. Okay, so alopecia, androgenic alopecia, this is also called as male pattern alopecia, and alopecia areata. This is formation of basically circular patches in several parts of the scalp. It is considered as alopecia areata. Alopecia totalis, that is total loss of scalp hair, and alopecia universalis, total loss of hair from the entire body. Okay, so these are 4 types of alopecia, and androgenic alopecia, male pattern alopecia it is called, which is one of the most prevalent form of alopecia and characterized by progressive miniaturization of hair follicles, lengthening of the resting phase, that is telogen phase of the hair cycle, and replacement of terminal hairs with vellus hairs. So moving next to next slide, alopecia. The mechanism behind these changes, which is not very well defined, we can say, but there are few studies which has pointed out the involvement of androgens, especially the hormone dihydrotestosterone, in the pathology of androgenetic alopecia, alopecia areata, which is another common form of alopecia, and it was reported to be autoimmune nature, and infiltration of CD4 plus and CD8 plus T cells and Th1 cytokine, and the presence of these. Okay, so this is I'll let with the help of this diagram, please understand that how androgenetic alopecia it occurs basically. So when poor blood flow to scalp, it leads to the nutritional deficiency of vitamin D, vitamin C, E, amino acids. Okay, and there what happens, the testosterone, the 5-alpha reductase 5-alpha reductase MON, which is responsible for conversion of testosterone to dihydrotestosterone, and there it leads to the production of hair damaging protein. So it shortened anagen phase, and the person is having androgenic alopecia, it starts losing hair. Okay, so this is how androgenetic alopecia occurs, which is male pattern baldness. And now, what are factors that causes alopecia? Okay, there are many factors. One cannot generalize that the hair loss is because of a certain or defined factors. And it's very difficult also to discover the actual cause of hair loss, okay, in alopecia. So now it's difficult and it needs a lot of research. That's why I also mentioned in the this session previously that this topic is having a lot of scope, and it's not like we need a full set of laboratory and having so many facilities to do research on this topic, this and related kind of topic. Okay, we don't need much resources. Okay, so why I'm mentioning this? Because generally what happens, whatever topic we decide for our PG dissertation or our PhD topic, it's very important for us to look initially about what all resources you are having. And except few universities, departments, and if we leave few university departments in India, we don't have much facilities and resources to carry out research, especially for PG dissertation. Okay. So to do a good research in minimum resources, we can select a topic like this or some related topic or some plant-based topic. So it's very easy to plan and do research, and yeah, that too good research. Okay. Now, okay, so factor, we were discussing about the factor responsible for alopecia. Physical, emotional stress, maybe surgery, severe illness, nutritional changes, and emotional stress, which one can not like figure out that why the person is losing hair. Therapy, like cholesterol lowering drug, a person is taking some drug therapy, like for Parkinson's disease, for some anti-ulcer drugs, maybe taking anticoagulant, anti-arthritic drug, okay, anti-epileptic drug. So, and increased level of dihydrotestosterone, okay, which is the testosterone metabolite, shorten the hair cycle and progressively miniaturizes scalp follicle. Okay, this is a very important cause of hair loss in males especially. And genetic component to androgenetic alopecia exists, and excessive sebum production causes a high level of 5-alpha reductase enzyme, 5-alpha reductase enzyme, which is responsible for conversion of testosterone to dihydrotestosterone, as I mentioned in the previous slide. So this is also one of the reasons. And cardiovascular diseases, excessive LDL in cardiac patient gets converted to 5-alpha reductase enzyme, and then 5-alpha reductase enzyme it increases the formation of dihydrotestosterone from testosterone to dihydrotestosterone, and then leads to androgenetic alopecia. Okay, so the next is now moving to the remedial part of it. Okay, remedial part as in there are only 2 FDA approved drugs, minoxidil and finasteride. Okay, minoxidil, which is a vasodilator, and it's a cardiac drug basically, and finasteride. These are 2 marketed products which are available for treatment of alopecia. Okay, the market trend predicts that the global market of hair loss treatment production will grow with a compound annual growth rate of 7.2% over the forecast period of from 2018 to 24. Okay. However, the results are disappointing and are far behind the expectation, as most of the time these products are not backed by rational scientific explanation. Basically, minoxidil is not, it was not intended to be used for the treatment of alopecia, right? It was like a kind of de novo invention that what has happened, the hair growth property of minoxidil was discovered as a side effect of it. Okay, side effect. Basically, it's a vasodilator and it's used in hypertension. So this scarcity of effective drug in modern medicine and the low success rate of marketed product is also one of the reasons which has forced researchers to focus on the development of alternative treatment, that is synthetic treatment for synthesizing. Both are synthetic drugs, basically. If we are, so far we are not talking about the herbal treatment. Okay, so alternative strategies involves either the utilization of crude extract. Now we are moving to herbal part of it. Crude extract or individual molecular entities obtained from plant origin. So what I am trying to communicate? I am trying to communicate, okay, there are few well-defined plants having some plant constituents, phytoconstituent, basically phytoconstituents responsible for hair growth. Okay, but apart from the existing one, there is scope of lot many plants which and lot many phytoconstituents which are still out of reach. Okay, but the thing is, we need good research, and we need good research to find out by fractional analysis, by doing column. We can identify. We can first identify, then isolate. We can isolate that particular fraction or that particular component which is responsible for the hair growth. Okay, so alternative approach, that adopting herbal treatment for it, crude extract. Okay, directly crude extract can also be tried in vitro as well as for in vivo by selecting a suitable animal model and specific agent derived from plant sources. This is what I was talking about, phytoconstituent, specific agent, that is phytoconstituent derived from the plant sources, from plant extract. So first what we have to do, we have to separate the fraction and then isolation of component from that fraction. Okay. The disadvantage of adopting herbal some herbal plants, some herbal extract is if you go for collection of phytoconstituent which may be responsible for hair growth activity. Basically what happens, we get very less quantity of that component, which is and after doing a lot of exercise of isolating that particular plant phytoconstituent for 1 year or for 2 years, the amount of actual phytoconstituent which we get is very less and it is not sufficient. So this is one limitation, and the solution part of it is isolated, identified phytoconstituents are available. You can buy those phytoconstituents. Yes, of course, they have got little higher cost, but if you can manage if you can manage the affording of that cost by maybe with the help of some grant project, funded grant project. So this way you can also go this way for applying this alternative for your research. Okay. Now, anti-alopecia drug, as I discussed, finasteride and minoxidil. Finasteride, what it is? Finasteride, it is dihydrotestosterone suppressing 5-alpha reductase inhibitor. Okay, it decreases DHT serum level and stimulate hair growth. This is its mechanism of action. Minoxidil, which I also mentioned that it is oral antihypertensive drug. Minoxidil has vasodilator effect, and which being vasodilator, what it does, it increases the blood flow to the scalp. Okay, the mechanism of minoxidil is it increases blood flow to the scalp, thus allowing more nutrition and oxygen to reach to the hair follicles, which shows or stop the hair loss and promotes hair growth. This is what is their mechanism of action, both finasteride and they are, I think, minoxidil also is OTC product. You can easily go and buy minoxidil, 5% solution. It comes, I think, in 5% solution minoxidil. And other drugs, arginine, Amanaxil 1.5%, B5 and B6 vitamin, and SP94 peptide. So these are some other drugs, but they are not FDA approved. Only 2 drugs are FDA approved yet. So there is a lot of scope. And anti-alopecia treatment, other treatment, gene therapy for alopecia, which is, however, is only limited to the laboratory scale until, but some examples are like Sonic, Hedgehog, SSH gene delivered to C57BL/6 mice. Basically, these mice are genetically modified. Okay, using an adenovirus vector stimulated the hair shaft production and resulted in longer anagen phase. Okay, so C57BL/6 mice are mice produced by genetical engineering. These are genetically born bald mice. Okay, and plant-based gene therapy is one, and herbal treatment we have discussed, and this allopathic medicines, finasteride and minoxidil. Okay, so now moving to some other plant-based herbal remedies for alopecia. Let me, okay. Hibiscus, they are very well known. Hibiscus rosa sinensis, and the reference for this plant is Adirajan et al. 2003 reported the effect of hibiscus flower extract in improving the hair growth. And there is one more reference quoted, which I mentioned, Gore et al. In 2009, Leucoris, Glycyrrhiza glabra, the family of Fabaceae, and 2013, in 2014, Upadhyay et al. And Deb et al. Has mentioned Leucoris for the treatment of hair growth in Ryan. So, and Bringraj, I have mentioned the reference. If you want, you can just take a screenshot of your screen. And stinging nettle, Urtica diacia, Urticaceae family it belongs to. And in 2013 and 2014, there are 2 references which I am have used Urtica diacia for the treatment of alopecia. Amla, from traditionally we have been using amla, shikakai, and all these homemade remedies also. So amla, which is also used for hair growth, and you will find a lot of references if you will explore herbal hair growth remedies. You will find a huge number of latest references as well. Fenugreek, This is methi, basically normal methi and kasuri methi. There are two types of Trigonella phanerogaceum and Trigonella phanerogaceum, and there are two different species, fenugreek, Trigonella phanerogaceum, and Trigonella cordiculata. Okay, so these are two species of methi, which is also used for the treatment, because traditionally in घर में भी हम लोग मेथी का ऑयल, लाइक मेथी को सरसों के ऑयल में डाल करके उसको बहुत देर तक ऑयल में बॉईल करते हैं, उसके बाद हम उसकी ऑयलिंग करते हैं. So, and we have also used basically this is part of my PhD research, hair growth, and I used two plants, Artica diacia and this methi fenugreek, two varieties. Okay, what I did, I prepared hair oil and hair gel. Okay, two things. Gel I used by soaking the method which I used, soaking some methi seeds in aloe vera. First aloe vera gel I prepared by using aloe vera leaves, and then methi, I soaked methi for some and then those I dried those seeds, and those seeds I don't remember. I think it's about I think I did in 2008, 2009. So some sort of oil we prepared by using this technique, by using aloe vera. We used aloe vera gel, methi dana in aloe vera. We soaked those methi dana, dried them, and some percolated with some sort of oil. I think sarson ka tel and coconut oil, two oil. So this way, and in other formulation, I remember plant extract I used. Okay, so you can also use plant extract, and of course, you can search for good references to plan the strategy and framework for your research. Okay. Anyways, I'm moving ahead. Plant-based herbal medicines for alopecia. So these are biological name of plant. Okay, and I'm just reading out biological name of the plant: Pilocarpus jabarandi and Albicus plicatorius, Cascata reflexia, and Ossimum centimum. And there are some more plants: Trichosanthus cucumarina. Okay, these are some belongs to Rutiaceae, Thicaceae, Fabaceae family, Leguminosae. Okay, and the plant part, which I have mentioned, that which part of plant is used and their traditional name. You can recognize the plant by their traditional name: jabarandi, green tea, okay, amarbale, tobacco, tulsi. Next, animal model. This I wanted to discuss little about animal model in vivo, for in vivo testing of hair growth. Okay, mice model. The model name C57B1/6 mouse model, C3HHEJ mouse model. Okay, red. Vistar albino red generally we use, and hamster, rabbit. So these are some animal model which you can use, but very commonly mice model is used because, as all of you know, that we share around 97% of DNA similarities to the mice, and that's why this animal model is very, very commonly used even in other researches also, not only in hair growth. Some anti-inflammatory or anti-diabetic or whatever you take example, you will very easily you will find mice model. Okay, so common animal model they are, and you can plan the study, the in vivo study by using extract or by using phytoconstituent. Both, either way you can plan, but For using animal model, there are a few important things that by taking ethical approval from ethical team and protocol approval, because you need to approve the protocol from the local institutional animal ethical committee or whatever you name in your institution, and the institution must have approval from CPCSA guideline. As per the CPCSA guideline, they give approval for conducting such animal studies. Okay, and you need to approve the protocol. Protocol means the each and every single step you have to write it down in the form of a format or a whatever format if you are having, your institution is having, and if it is not having, like simply you need to propose that what you are going to do, how many animals you are going to use, how many animals you need to sacrifice. Okay, and there are some conditions associated that you have to first you have to go through from it, and then you can and even if your institution is not having this animal ethical committee and this facility, there are you can take help and from the first the sources, maybe your colleague, your friends, or other institution which is having first thing, or you have to arrange. And how you are going to arrange, it depends on the kind of collaboration you are having, or you just can inquire to other institutions if they allow you to conduct such studies. You can just go and send some one of the team research team member to that particular institution and conduct research for whatever time it takes. I think 2 months or maybe 3 months, not more than that. Okay. So I'm showing this some photographs of just a minute. Some I used mice model in vivo anti-alopecia. The photograph of rat treated with control, standard, and test. Okay, control we use only olive oil. Standard 2% because minoxidil. Yeah, I forgot to mention that minoxidil is also used as standard. 2% minoxidil solution is used as a standard for carrying out hair growth activity. Okay, and test herbal oil, as I mentioned that I prepared herbal oil. So on 5th day of study, there was hair growth, which you can understand with the help of this graph. Also, the time in days versus the hair growth initiation. Basically, 5th day, it was the initiation time, okay, of the herbal oil, which I prepared, and the plant which was used, the Trigonella, both the species, I think Trigonella corniculata and Trigonella fornicaceae. Okay, and this is in vivo hair growth study showing hair growth initiation day, basically photograph. Okay, control, standard, you can see T12 and T21. These are sample name. At 6th day, T1 and T2. Okay, as you can see, the hair growth initiation time was less in test sample as compared to the standard, as that too as compared to standard and control. And this we went for biopsy of few samples, or biopsy of hair follicle of albino rats after treatment of 1 month. Okay, so we selected one sample for biopsy. The one we like selected one sample that shows the best results among whatever the test samples were. Okay, so biopsy, you can see the test, standard, and control. Control is normal. Here you can consider the hair follicle, the picture or the biopsy of normal hair follicle, standard, with the treatment of minoxidil, and test one. Okay, so that's all this I wanted to share from my research. And yeah, one more thing is remaining. Let me complete. Common in vivo experiment design area in anti-alopecia research, depilation model, and dihydrotestosterone induced alopecia model. These two, depilation model, it involves removal of hairs from animal model either by shaving or by using hair removal cream. Either a patch is shaved or the whole back or the body of animal is completely removed. And second one is dihydrotestosterone induced animal model. So either these are two types, and very commonly you can use mice model or rat model, whatever is available, and if you can and depending on the plant and number of samples you are having, you can plan in vivo studies. And for in vitro we are not going to discuss much about in vitro hair cell culture. Basically, you'll also find many references if you will explore about in vitro hair growth, hair cell culture, basically the techniques of hair cell culture. So that's all, and it was a little part of my PhD research also I discuss and share with you. So these are, and lastly, just a minute. First, I'll stop sharing, and if you have any queries first regarding this topic.
Thank you, ma'am, for such knowledgeable session. We are very privileged to have you today. Now, anyone have any question he or she may ask? Ma'am, there is a question from KK Srivatsava. Can alopecia be genetic?
Yes.
Can alopecia be genetic? Genetic.
Genetic, yes, of course. This is one of the factors, one of the reason for which I mentioned that there may be many reasons for alopecia. So this is one of the reason and very important reason of genetically. You will find so many examples around you also that the father is, if father has lost hairs at the age of 50 or 60, and so the son will also lose.
Okay. Thank you, ma'am. Thank you so much. Now I would request to Dr. Shweta, Assistant Professor, School of Pharmacy, Arka Jain University, to kindly felicitate Mona ma'am for her wonderful session. Thank you, ma'am.
So dear participants, one more thing I want to mention regarding our MOOC on industrial pharmacy. The enrollments are open. So this is the only pharmacy MOOC, industrial pharmacy 5th semester. And it is also for 7, I think for 5th, 6th, 7th, 8th semester students. And if they are faculty members, I request all the faculty members to let students know about industrial pharmacy MOOC, and enrollments are open. You can enroll yourself, and so is for academic writing MOOC. You can visit swayam.gov.in. You will get both our courses there. So please do enroll to the course. And I wanted to mention this. I don't have right now the link, but I don't have. Just a minute. If I'll share in chat box. or I'll share to Dr. Dean sir, and he will share with all of you. Okay, must be having.
Thank you, ma'am. Thank you so much. Now
Just
moving.
go to, just go to Swayam, swayam.gov.in. In search bar, if you'll type academic writing or industrial pharmacy MOOC, you will get it. Okay.
Okay. Sure, ma'am.
Yeah. Thank
Thank you, ma'am.
Thank you so much. Thank you, Arka Jain University, for giving me opportunity to interact and to discuss this topic with all of you. And thank you, Dean sir, for invitation, and thank you, dear participants, for listening, for patient listening.
Thank you, ma'am. Now moving ahead for our next session, we have Professor Fatima May R. Tesro, Dean of Pharmacy in San Pedro, the Philippines. Welcome you, ma'am.
Thank you so much. Is my slide seen by everyone now?
Ma'am, please let me give you some introduction about yourself.
Okay.
She is currently the Dean of Pharmacy in San Pedro College. She is an educator for 18 years handling subjects in biological and pharmaceutical sciences. She was the recipient of 2070 PERA Outstanding Educator of the Philippines. Ma'am has various awards in her credits. With these words, I welcome you, ma'am. Please kindly enlighten the session. Over to you, ma'am.
Thank you so much. Am I audible? Can you hear me from your end?
Yes, ma'am. Yes, ma'am. You are audible.
Thank you. Okay. In the interest of time, I'll be making my presentation quick as we are actually way late from our expected run. Okay, so good afternoon, dear participants. Allow me to appreciate and commend the organizers of this forum, this conference. Thank you so much for the invitation. So I'll be presenting the current trends in extraction and screening of pharmaceutical herbal plants. Now, majority of us would like to look at pharmaceutical and herbaceutical or cosmeceutical as one of the most important commodity as of the moment. So the context of my presentation today is the current scenario that we have in the Philippines with regards to extraction and screening of pharmaceuticals that are plant-based and herbaceutical products. And on some areas, it is on the parlance of the Association of Southeast Asian Nation Landscape. So here, if you can see in the presentation, it is looking back 5 years ago. This was 2016. So the illustration is showing us a global herbal market size. So if you notice, when we speak of herbal preparation, we usually look at it as a preparation that are made up of tablets and capsules and powders. But if you notice from 2019 to 2021, you can really see that there has been an increase. This purple arrow here, there is a tremendous increase in the utilization of extracts. So these extracts come in various products. It could be formulated as oils. The one that was mentioned a while ago, it could be used for cosmetic purposes, for the application in the hair, or rubs, liniments. So these are some of the products that are derived from extracts. And of course, there are also oral preparations that are under the food supplement category that are marketed and are derived from extracts. So if you notice from the diagram, you could see that there is an increase in the consumption of herbal medicine, and usually they come from powders, tablets, capsules, and extracts. Imagine 2016, that's just 5 years ago, and the consumption is 71.19 billion. Okay, this is in the works of 2009, Anish, Hisham, Shakur, and Madhu. So from that, we can see that there is wide use of herbal medicine, complementary medicine in developed countries. So in the past, only developing countries has an increased consumption of these herbaceuticals and pharmaceutical products that are derived from plants. But today, there is a clamor for the use of these products, not only in developing countries, but in developed countries as well. As a matter of fact, if you look at the current 2021 release, so this was actually released in Express Wire, dated September 6, 2021. There is a global herbal medicine market survey. It is a detailed forecast that made use of SWOT and value chain analysis to utilize the gauge of the market. And it says there that herbal medicine and botanical medicine or phytomedicine that are derived from plant seeds, berries, roots, barks, flowers, and for medicinal purpose are now gaining popularity. And this is the forecast of the Market Watch. It says that probably 116 million by the end of 2027. 2027 would be the market of extractives or extracts. And according to this study also, herbal medicine would fatten the market size to 150,270 million in the year 2021. So there is a 6.3 variance or increase from 2021 to probably 2027. And along that line also, if you watch and read in this particular article, the clamor for the use of herbal medicine, particularly extracts, oils, liniments, those that are aromatic in nature, volatile in nature, has been increasing. The clamor for that has been increasing, particularly in this COVID pandemic, meaning people are going back to natural sources for healing. So on this note, if you notice, this is a diagram showing to us that these products are a testament as to how people now are trying to view traditional medicine. So traditional medicine today are viewed as naturally occurring and they are plant-derived with minimal or no industrial processing. And usually it is used for the treatment of illnesses, local or regional. So we have different practices. You have Ayurvedic practices in India. We also have Filipino traditional medicine practices, and along with other herbal medicine practices in the Asia Pacific. And there is an increase in the market share for that. So why am I placing a cross here? Because some of these products are posted by the Food and Drug Authority, because these products would like to claim that they are natural, they are derived from plants. Unfortunately, they did not underwent stringent quality control processes. In short, there was no screening as to the quality of the product. And another thing that we have to look into is that, are they really the extracts that we expected them to elicit activity? And so all of these are FDA advisories that these products are not registered. And at that point, you would know from the scenario that I presented that there is an increase in the herbal medicine market. It's a strength because basically people are now aware that we can get and source again healing and medicine from nature. As Hippocrates would say, nature is the best physician. So that's a strength. And while looking at that strength, looking at the scenario, you could also see, today there is a clamor for traditional medicine, but the problem is there is still the use of traditional method of extraction. And we know for a fact that when we use these traditional method, I think all of our pharmacy students can attest here, there are some disadvantages such as impracticality because of the wastage. One, you waste a lot of solvent and that can be non-environment friendly, meaning it can harm the environment because definitely we are extracting polar constituents using polar solvents. And we know for a fact that sometimes these can be wastage as we cannot recover them, or there is poor recovery in the traditional method. And quality in terms of extraction and screening is compromised with the use of the conventional method. I'm not saying that conventional methods are not good. The principle of conventional method is there and it's for our students to learn, but there has to be an upgrade of the things that we do so that we ensure that the product that we are releasing in the market, its value is not compromised. And so the threat there is, of course, the expenditures. We are using current trends. No, I will be introducing what are the current trends in screening and extraction that we have to imbibe in our operations, in the academe, in research, and in the industry. Industry are well far ahead because they are using already technology-based extractives and technology-based instruments for screening. But we also have to have an upkeep here in our respective institutions. And we have to ensure that our students are trained well in the manner of the use of conventional, but more on the use of the current instrumentations and methods. Okay, so the opportunity now is that we have more and more and more technology using microwave assisted techs. So these microwave assisted technologies are actually greener in nature. I say greener not because we just saved sample, meaning it utilizes just very small amount of sample, but very high amount of yield. I also say it's a green technology because we are trying to avoid those methods that will utilize organic solvents that could already be harmful to the one handling and harmful to the environment. So it's good to start early in the training of the students, expose them to these sophisticated instruments, and two procedures, screening and sample preparation prior to screening needs to have updated procedures. Yes, I understand we are already exposed to ICP, inductively coupled plasma emission spectrometry. We are exposed to flame photometry, furnace, using flame or furnace, and etc. in the analysis of heavy metals. But we also have to ensure that when we screen them and sample prepare them, even the sample preparation should involve a greener technology, which uses microwave assisted technology. Just a disclaimer, I might be posting photos of a particular product or instrument, but that does not mean that I am promoting this instrument. It's just that that is the instrument we are currently using in the institution where I am. Okay? So those are the things that we have to consider. And these are also one of the things that we have to ponder on. Okay, and that is presence of toxic metabolites. Whenever we talk about botanical medicine or whenever we talk about phytomedicine that is derived from plants, automatically what is in our mind is its pharmacological effect, its biological constituent. What we don't think of is that there could be other excipients, other constituents present in that plant that is no longer safe. And that is the plant metabolites, the toxic metabolites. So one of the things that we are to be careful of is the presence of this heavy metal. Because definitely when we promote phytomedicine, we are also promoting longevity. And part of longevity is that they can be consuming these phytomedicines for a long period of time. So there is a chronic exposure to it. And so given that, if heavy metals are not screened in these herbal products, the tendency would be our patients would be consuming these toxic heavy metals. And so as Hina et al. Would like to present in 2011, going advanced in research, it is found that plants not only contain toxic secondary metabolites, because there are also plant metabolites that are toxic, but they also have contaminants that are environmental pollutants, and especially heavy metals, which on long exposure can lead to serious health issues. So this is what I'm talking about. So again, when we look back on the biological activity of the bioactive components of plants, that's good because we are trying to target a specific illness and we are trying to target what specific plant is good for that, what specific bioactive metabolite is good for that. But along the process of making it, we have to ensure also that there are no contaminants, no heavy metals, We screen these things because they can also pose serious danger to the health of the one who will be consuming it. So again, balms, oils, camphorated products. Sometimes we market that this particular balm has a presence of oil. The oil is coming from this particular extract. But there is a question, is it really the right extract? Did it underwent correct extraction procedures? Does it mean that we just evaporated this particular plant and we got this particular residue that is already considered an extract? These and many others are issues that we also have to address. So here, I would like to present to everyone the two important things that we have to consider in the discovery of pharmaceutical products. Our topic is natural and novel pharmaceutical discoveries. And when we look at these two natural and novel discoveries, we should always see to it that the screening and extraction are done right. No, not just to secure that there is an activity, but also the way it was processed, it has to be screened well, and the extraction, it has to be extracted well. So these two are critical. So to proceed, I would like to begin with screening. Why do we screen at start? No, I firmly believe the screening should start when we harvest the plants for production. Okay, so it begins with the raw material, and it should use accurate method of screening and the correct instrumentation. Why do I say that later? I will tell you the current trends on screening this, new instruments on screening this, and why is it that we try to deviate from the conventional methods already. So the use of current sample preparation instrument is advantageous, and the use of highly sensitive method and instrument yields accurate results. So these are the two reasons why we have to ensure that we use the current trends. So begin with an end in mind. The end in mind is to make a product that is safe. And so we have to secure that the moment we have the raw material, it has to be that that raw material should be free, no, from contaminants like heavy metals. And we know for a fact that elemental, what are the heavy metals that are usually contaminants in the soil or in the atmosphere, in the air? We have mercury, or we have heavy metals like cadmium, or arsenic, or lead. So all of these four metals are the four heavy metals that should not be consumed chronically when a person is taking a herbal product. Why? Because these are elemental in nature, and we know for a fact that these elements cannot be destroyed. And so once they are present from the raw material, definitely, even if it undergoes process, it will still be the same mercury, the same arsenic, the same lead. So why? Because they are high density, they are essential, and then non-essential. So meaning we have to ensure that it does not pose toxicity, okay, at a specific level, because sometimes some of them are toxic even in a minute concentration. We know for a fact that parts per million, like greater than 10 parts per million of lead is already dangerous, or greater than 1 part per million of mercury in consumption could already be dangerous orally. So we have to secure this. Okay. So how do we do that? No, why do we do that? Prolonged exposure to heavy metals may cause adverse effect, no, and toxicity due to the capability of the heavy metal to bioaccumulate and may disrupt the functions of the vital organs. And usually it's the kidney and the liver that is affected, and the brain. Okay, this is according to AHPC. And because of that, those are the whys. We have to secure that mercury, lead, arsenic, and cadmium should be in a very minute amount, or as much as possible. We should screen these raw materials so that they are not present in the herbals that we are trying to process. And the very first current trend today is begin with sample preparation. We know for a fact that when we try to test heavy metals, you know this is part of your instrumentation lesson. How do we assess heavy metal? We make use of what instrument? AAS, atomic absorption spectrophotometry. That's the standard method of analyzing heavy metals. But prior to analyzing that, we have to be serious with the pre-analytical step, meaning the step prior to AAS, and that is sample preparation. When we prepare the sample, when we prepare the herbal product. Later, I will tell you one of the outputs of the research I had on Davao River. That is where I am situated, and that is where majority of the herbal plants are being secured and harvested. In the past, there has been studies on it, and then they tried to test it, and it says that it does not contain heavy metal. But they made use of the traditional method of testing it. So it's actually the method that became erroneous, because when I tested it using the current protocol, using the current method, using current technology, using microwave assisted digestion, then the recovery is high. When it was tested using AAS, it showed that the mercury and the lead are way beyond the allowable permissible unit. So what does that mean? It can pose danger to the health if we continue to be using the conventional way of pre-analytical testing. So that's the one. So we use the digestion of solutions. We know that prior to testing, we test them. So what are the sources of the errors of the analysis? So there exist supplements and herbal medications that underwent AAS tests, and they passed the detectable limit. They passed the limit. But when they are tested again, prior to retesting it, they check what method was that, and then the method was traditional wet-dry method. But when they tested it again using microwave extraction, using microwave assisted technology, there they found out that there was really high amounts of lead. Okay, so that is where error in analysis comes in. Okay, and these errors should be preventable. Look at this. This is a diagram in the study on analytical methods 2013. These are the causes of error in chemical analysis of herbal plants. And number one is the sample preparation, meaning how they were prepared. And this was using the conventional way. I'm sure you know the hot plate manner. You have the sample, let's say bark of a plant, you immersed or you triturate it, comminute it, and place it in a solution using an acid digestion. You could use nitric acid and a combination of perchloric acid or nitric acid and hydrofluoric acid, depending on the cookbook or whatever is the combination of the acid that is used to digest the sample. And then heat it over a hot plate, and sometimes you have to subject it to a very extreme temperature. Sometimes the beaker will break, so the tendency would be you will recover only a very small amount of the smoke or the flame or the residue or the precipitate. And then when you test it using AAS, the AAS result will say very small amount of lead. But actually it's not that lead was not there. It's just that your process was the one that is erroneous because you made use of the conventional method. So that's that. So this is a research that really shows that there is really an error in the sample preparation, and the rest are other errors. But majority is really from sample preparation. So what does that speak of? The bottleneck is the sample preparation. So either we use two tests. It could be flame photometry, or we can use furnace method in our atomic absorption spectrometry, or we use the ICP method. But again, the one that is crucial is the sample preparation. So regardless, whatever your sample is, it could be from nature, herbal medicine, or other things that you would like to assay, it has to be that you have a very good technology in the sample preparation prior to analysis. No, Modern analytical equipment gives better accuracy, okay? And analysis may be done in few minutes and seconds. Whereas if you compare it to conventional sample preparation, time, reagent consumption, and handling can really be a factor. It will play, okay? So that is my point there. Now, talking about that, so what are the current trends, no, in sample analysis? So to trace heavy metals, to trace them, analytical measurement is as good as the sample preparation itself, meaning looking to trace analysis puts more demand on the sample preparation. Ito yung kulang. This is the one that is missing, no, in all the testing that's done. A lot of laboratories, a lot of researches done by students, done by PhD students here in the Philippines and even outside. You check it, there is a systematic study on this that they say that these herbal products that are made are really of good quality, but when tested using AAS, it also yield a very good result. But when you look back at the method, the very good result was just because they made use of the conventional method. But when they were retested using a good pre-analytical instrument, then they were able to detect mercury, lead, and arsenic present in the sample. So I now say that the more appropriate term for sample preparation is pre-analytical step, because really the correct way of doing the process or preparing your substance before analysis is critical in making an accurate analysis. So we are used to this. This is the traditional, conventional method, the hot plate digestion. Well, the advantage is it's cheap at start, but in the long run, it will not be cheap because you have to be replacing your hot plates. You have to be replacing your beakers, your apparatuses. You have to replace them from time to time because they are exposed to extreme temperature and definitely breakage will happen. The disadvantage is number one, health hazard of the one handling it, the one performing the analysis. Our students, us, pharmacists in the quality control, that could be a health hazard. Why? Because definitely if it's the traditional method, we will be exposed to acid fumes. Okay, time consuming, limited productivity. Why do I say limited productivity? Just imagine you have to have pre-analytical steps, sample preparation of, let's say 6 hours, 3 to 6 hours before you can get a residue before you test it in the AAS. And you will be consuming so much acid, which is hazardous to the environment too. And then there is low to moderate digestion quality. You are not digesting it right. Okay, so that's for the hot plate method. Just see that. This is an illustration, image courtesy of this particular blog spot on the different acid digestion setups that they have over a hot plate. And I tell you, it may look simple, but you will be watching this every now and then because breakages will happen simply because you are exposing the acid to extreme heat. Okay, and the problem is you are actually after the residue on top of this hot plate, of this cover glass. Okay, and when you do that, sometimes you cannot recover so much. And if you cannot recover so much, it will affect your analysis. So there is now a new system, the current trend, and that is a closed vessel microwave. And it has to be regulated. I think this is the one that is lacking. There has to be a regulation that the pre-analytical step should utilize this because this is critical. So the closed vessel microwave system makes use of better digestion. It's fast. At the same time, it's safe. It's easy to use. And there is a full control of temperature and pressure. And the digestion quality is supreme. So this closed vessel system, okay, may increase application performance, very good throughput, ease of use, and there is reliability. So the ROI will be coming better. Okay, so these are things kasi to consider in the sample preparation or pre-analytical step. And we have to see to it that these are met. And when you use a closed microwave system, all of these are well satisfied. So the best choice is to merge the application with the system features. So quality analysis starts with great sample preparation. If we have poor sample preparation, there is poor digestion, definitely the results will be affected. And so the use of a microwave technology, particularly a closed vessel microwave system, will answer to that. Okay, now how does it answer? Definitely it has a powerful temperature control. So temperature, as we all know, is one of the drivers of digestion. So you will be subjecting your sample to a very high temperature, but here using a microwave system, the microwave increases the temperature without affecting the vessel, because also the system has a Teflon vessel that is able to withstand extreme temperature, okay, and extreme pressure. Okay, so it depends. There is a rotor, no? There is a rotor. How many rotors? In the academe, we only have few rotors, no? Pwedeng 8. The most important thing is that all your students are exposed to how it is being done, the acid digestion. Well, definitely not as of this time because it's a pandemic, but hopefully when restrictions will ease out, then they should be exposed here. Okay, so look at how we rated the different approaches to sample preparation prior to atomic absorption spectrophotometry. And this is the comparison. Conventional method, well, it's really poor because it makes use of radiation. So from the hot plate, it will radiate to the vessel before it can heat up the acid with the sample. So definitely sometimes the acid would require very high temperature and the vessel can no longer withstand the heat, it will break. So this is the stars, or how it's evaluated or appraised. The in situ method, the only problem in situ method is control in all vessels. You have to open it up and then close it again and adjust the vessel. But here, using our milestone, or actually using any microwave assisted system, then the ease of use, direct control, the temperature control, the safetiness is being addressed well. Okay. So this is the mechanism I've been telling everyone about. How come it has a very good control system, hindi puputok. Unlike in the regular setup, the tendency would be there will be explosion of the vessel, breakage of the vessel, or if there is an increased pressure, there will be explosion that will happen. All of these disadvantages are eliminated with this technology, because this technology has a vent and reseal technology. So definitely the sample is inside here, and then automatically it will be exposed to heat. The rotor will keep on moving. Sometimes due to the extreme temperature and elevated pressure, the tendency would be that the content inside might explode. But the very good thing with this technology is that it will vent out. So it will automatically, at a certain temperature, it has a temperature control and a pressure control. So it will have a detect. So meaning it has an intelligence system that will detect extreme pressure and extreme temperature. And once it detects that, the tendency would be it will also regulate by opening a certain degree or amount of the vent so that a little of the air goes out. And then after a while, if pressure is already reduced, it will reseal. So it has a vent and reseal technology. So it ensures full recovery of your product because there's no wastage, no breakage whatsoever. You also did not open and close it again, because once you open it, it will evaporate. It's highly volatile, so no loss of volatiles, and it's safe. Okay. So with that, this is the classic setup that you can have in the lab. So you have here your digestion system. Okay, so this is where you digest sample. This is where you set up and lock and seal all of those Teflon containers and then place them here, and then voila. 30 minutes, you just have to wait. After 30 minutes, it's done. But if you use the conventional method, well, you have to wait hours before you can get it, and you cannot, and at times after 3 hours, you cannot recover a single precipitate. So that would be a problem. So all of these, nutraceuticals, agriculture, environmental, food, chemicals, even pharmaceutical products and pigments can all be digested using a microwave assisted digestion setup. Okay, and these are the different accomplishments of targeting heavy metals and other elements that you would like to assay. And the sample preparation or the pre-analytical step that you use makes use of a microwave assisted technology. And this is my point. Now, regulatory requirements for safety assessment on herbal medicine, this is on up to 2012, okay? They have same requirement as the conventional pharmaceuticals. And there is little special regulatory requirements and no requirements at all. So this is my point. There is still a lot of countries with no specific regulations on this. And if there is no specific regulations, probably the industry, the academe, the institution will just use the regular conventional method. I'm not saying that it's not good, but all I'm saying is that it will affect the result. Okay? And so it will have a lot of impact. So these are the current US standards for permissible limits. But again, these permissible limits will just be numbers. If at start we do not begin with the correct procedure of screening our plant and raw materials. Okay. So now after we sample prepare them, now we will really screen them using instruments. So before we use colorimetric method, but we are not so much on that now. I will focus on the instrumental method. So these are the current trends. You have your furnace, flame atomic absorption spectrometry. You have your graphite furnace atomic absorption spectrometry, and then you have your inductively coupled plasma emission spectrometry. And then you have your XRF or X-ray fluorescence spectrometry. So these are the current trends in analyzing heavy metals to screen your herbal products. Okay. So, well, in colorimetric, there is still a lot of steps, like you decarbonize, you digest, and then you add a hydrogen peroxide before you can proceed with the analysis. Okay, but these are some of the method. This FAAS is actually an old method, but still it has its perks even today. It's not that expensive, and yet it will provide you an accurate result. Okay, so that's me doing the flame atomic absorption spectrometry. This is the flame. This is one of the oldest technique. And if you will compare flame atomic versus graphite furnace, most likely there is just a deviation of 1% from the result. But the principle involved is still the same. You have a hallowed cathode lamp, you have a flame, you have an atomic slip. Okay, so from your sample, okay, it will be nebulized and atomized, and that sample will move through the flame. And then the lamp will bombard that flame, and then your monochrometer splits it and detects the area of absorption. And from the absorption, it will now detect the amount of iron, the amount of metal that is present in the sample. Now, how is this different? Your flame absorption spectrometry different from graphite furnace? It will only be different in the source. Because here you make use of a live flame source, usually making use of nitrous oxide and acetylene gas. But in your graphite furnace, well, it's graphite that is used as your source. So your GFAS makes use of the graphite furnace. So in the regular flame atomic, this is the flame, but here, this is the graphite furnace. But the same principle is being observed. You have a light source or a hollow cathode lamp. Then you have a sample injector where yung na-acid digest, the one that you acid digest, no, the one that you made use of a microwave system is the one that is introduced here. And then a monochromator will concentrate that light, and then it will be detected, and then you will see the results in your decoder. Okay, and then you have your ICP, MS, pwedeng mass spectro, or ICP is inductively coupled plasma emission spectroscopy. So here you make use of ICP, plasma, you make use of plasma in elemental analysis of the ions. No? The ions generated by the sample is measured using ICP. And then this is one of the newest trends, the X-ray fluorescent spectroscoper. Para lang siyang gun. It looks like a gun where you just point it out to a sample or an herbal product, an herbal rub, an oil. You just point it down and automatically it will project here in the graph at the interface the amount of heavy metal present. Okay, here, this is not as accurate, no? But I would like to tell you there is +1, -1 deviation. Unlike in the graphite furnace, ICP, and flame atomic, that it really will provide you a relatively accurate concentration of the heavy metal that is present in your sample. So that's the manner how XRD is being done. Actually, I tried it in my own ring. I tried to flash the X-ray diffraction, and you can really also see the amount of gold present in it, as gold is also a metal. Okay. So I will not deal with that in the interest of time because I'll be proceeding to extraction. So my point here is that trace metal analysis is a crucial analysis in herbal preparations. We do not just test bioactive components, but we also test and see to it that it is toxicant-free. And to ensure that, we make use of good screening methods, and good screening methods will use either flame atomic spectroscopy, graphite furnace, ICP, or your XRD. But before you proceed to all of these four or to any of these four, you have to ensure that your pre-analytical step also makes use of the current microwave-assisted technology so that you can really digest well your sample before it is being screened and tested. So this is what I've been telling you. We had a study here in San Pedro College. We have this testing of all the herbal plants along the coastline of our Davao River, because majority of the native dwellers here would source out their medicine from this area also. Then initially, there was a test that was done using conventional method, and the results of the test is that none of all these plants contains lead and mercury. But when it was retested, this time using an acid digestion method using microwave assisted technology, START-D technology, then there we found out that a lot of the plants really have heavy metals. So these were some of the plants that were tested and detected that there were really heavy metals. So just imagine if all of these were made into products and people are consuming it without necessarily testing the toxicant level or the heavy metal, okay, present in these plants. Okay, so even plant-based cosmetics are not exempted. Whitening cosmetics has to be tested with mercury, and so plants also has to be tested with absence of mercury at start. Okay, so now I'll proceed to extraction. So in our institution, we have this instrument. We use, we call this Ethos X. Ethos X is a technology that is under microwave assisted microwave assisted extraction. It is also termed as solvent-free extraction. Why do we say solvent-free? Because the regular extraction that we all have and we all know is that it always involves either petroleum ether, or it involves hexane or an alcohol, or whatever solvents, polar or non-polar, that is used to extract components of plants. But here we are extracting essential oils, and the way terpenes, monoterpenes, we are extracting them in a manner where we only used the plant, immerse them in water, but discard the water eventually, so it's solvent-free, and then test it here in this particular technology. So this is much more better, an accurate method and an accurate instrument. And yes, it is a green technology because you are doing away with the use of very toxic solvents. So that's that. Now we know the conventional method. I don't need to expound all of these things, but these are some of the conventional method that we use. Perhaps we are still exposed to Sochlet extraction so that we can extract oils, essential oils and volatile oils. Okay, but I would like to introduce that these methods have drawbacks. Number one, it's time consuming. It would require a lot of amount of solvent, difficult in solvent recovery, and sometimes thermolabile components, especially yung mga volatile oils. Majority of your terpenes, monoterpenes are highly volatile. Majority of your essences are highly volatile, and some of these essences are very useful for therapy, but then all of them are destroyed because you made use of the traditional method, and some of the thermolabile components are already destroyed. So the integrity of your product is already compromised. And so to avoid that, there's a new technology using this. You can use microwave assisted extraction, ultrasound assisted, using supercritical fluid, accelerated fluid extraction. But I would like to focus more on microwave assisted extraction. So it makes the one that we use here in the Philippines, particularly in San Pedro College, as we are the first to have this instrument, is Ethos X. So it represents a state-of-the-art, still a closed vessel microwave system with a very good design, a superior vessel, we call it the reactor, that has the capacity to withstand extreme pressure and extreme temperature, and it provides us with the highest yield. Okay, so this is primarily how the system works. Okay, in this particular system. So in the diagram, you see three illustrations. So the first illustration there actually is simply the glands. Okay, so the diagram will show you the cell, the plant cell. Okay, and in that plant cell, basically, the oleferous glands are still intact. The oleferous or oil receptacles are still intact. But using microwave heat, using magnetron that is produced by the microwave system, the microwave actually increases the solubility of the analyte of interest, okay, by giving a more precise and accurate result. So using that system, okay, it takes advantage on internal heating. So here in the second diagram, the microwave produces, the system gives microwave selective heating. So that particular system now will provide an internal heat to water, to a very small amount of water that could be present inside the plant or just present outside the plant as a moisture. So it will provide internal heating to hot water, and then your sample will begin to distend. So it will distend its cells, it will rupture the glands and the oleferous receptacle. And on the third diagram, it will show you now that it frees the essential oil and later is evaporated in situ and then goes to the condenser. Okay, so that is the principle of the analysis. Okay, so this actually is the feature. This is actually one of the setup. I don't know what, okay, this video will show you that there is an interface. So this is the instrument. Inside the instrument is a reactor chamber that is containing oregano. So the one in the beaker are the oils that I was able to extract for 45 minutes, that short. And then you notice here in the diagram, there I usually have enough room to adjust temperatures, to adjust pressure, and I can see it real time. No, the good thing here is there is also an app that you can place it on your phone. And so whenever you move to another area, another room in the laboratory, you can just leave it there and then use your mobile phone as your monitor. Okay, it will also give you some real-time analysis. So that's that. So the system may work via gravimetric method if you are after flavors or if you are after for constituents that can be extracted by gravity. So a good example here is if you are after for, this is I think oil or ginger, this is ginger, and you wanted to get important components of ginger that is used for, there is a current study on that, on ginger, in its capacity to help prevent the docking of the virus in the lungs. So you can have, you can use this setup, the flavor extraction setup. So your condenser is down below, and then this is where you collect your oils. Here is the classic setup for the fragrance. Me, along with Professor Kerikinsha and Professor Agustin, are into a research right now. We are trying to come up with a product. I cannot divulge the complete details yet, but we are using the same instrument so that we can extract the oil, and we hope to test the oil on a lot of bioactive components. And there is one component kasi, there is one, the plant that we are studying actually is a well-known plant, but there has been an issue that this plant is actually poor bioavailability because of its poor solubility. Because majority of the extractions that were used were that they extract it using different solvents, and then the tendency is it will just leave the system, our system, without being dissolved. So now our study is more on getting its volatile oil, as it is the one that has better effect and more bioactive components that are present. So we were able to use this instrument and we got a lot of yield. Okay, the yield was really very good. So this system, the microwave assisted system, addresses the limitation of our 90-year-old technology. You know what's our 90-year-old technology? Using the Clevenger method. Even today, standard procedure is still based on a Clevenger method. And this dates back to 1928. While yes, Clevenger method is classical, it's also still working, but sometimes the yield is compromised. So the yield is compromised, and then at the same time, it is not a green technology. So here we used a solvent-less, fast extraction green technology with no thermal degradation. So these are examples of some of the yield. So citrus peel, garlic, ginger, lavender. Imagine this is just the sample amount, 7 to 5 kilograms, and yet the yield is very high. You cannot get this yield. No, this is a dramatic result of the yield if you use the regular Clevenger method, and it would take you hours for a Clevenger, when in fact here you can just use it up to 45 minutes. You can time it up to 45 minutes, similar to this one. So this is one of the things that I would like to tell you. If you want to get very good essential oil with ginger, if you want to at least get 4% of the yield plus some of its phenolics like gingerols and shogaols and total carbohydrates, then the MHG, the microwave gravity method of your microwave assisted technology is a very good and powerful way of extracting it. You can get this high yield even with a 1 kilogram sample. So that's that. Those are the research that we are currently doing in SPC. And we are incorporating it in our drug discovery. And this is part of the services that our San Pedro College Animal Laboratory and Research Hub is doing. By the way, we are extending our laboratory to those who would like to have their samples tested, samples processed at a reasonable price, as we are maximizing our instrument. I would like to end my presentation with this, that safety is from womb to tomb. We do not compromise the safety by coming up with just a very good product. A very good product means that it is pharmacologically good, but it should also be toxicant free. And when we speak of safety, it is from harvesting to screening, to extracting, and to dispensing. Thank you so much for listening, and God bless everyone. Mabuhay.
Sir. Thank you, ma'am, for this wonderful session. It was very informative for us. Now, may I request to Dr. Babita Sarangi, Associate Professor, School of Pharmacy, Arkajan University, to say few lines to appreciate Professor Fatima for her contribution in the plenary session. Over to you, ma'am.
Good afternoon, everyone. Audible? Sorry. Good afternoon, everyone. Gratitude is the best attitude. With this, I would like to take the opportunity to thank our distinguished speaker, Professor Fatima, Pharmacy Department, San Pedro College, Philippines. Thank you, ma'am, for sharing your knowledge and experience. It was quite informative and thought-provoking. Thank you for taking time from your busy schedule to join the session and to grace the occasion. Thank you. Thank you on behalf of Arkajan University also. Thank you, ma'am. Once again, thank you.
Thank you, ma'am. Now, this brings us to the last plenary session of this day. I would like to welcome Mr. K. Shravana Kumar. Sir is a postgraduate in pharmacy, having about 25 years of experience in pharmaceutical industry. He is graduated from Madurai Medical College and postgraduate from BITS Pilani. He started his career as research scientist in TTK Pharma Limited, Chennai, followed by his assignment as development scientist with Dabur Research Foundation, New Delhi. With his experience in validation and technology transfer as a part of research and development, he had expanded his career into the field of quality assurance in Force India Laboratory Private Limited, Chennai, which is a fast-growing pharmaceutical company in India. Currently, he is Associate Vice President Corporate Quality in Force. We welcome you, sir, for today's conference. Now, may I request you to please kindly lighten the session with your knowledge. Over to you, sir.
Thank you, madam. Thank you, madam, for the introduction of me. And I can share the screen, right? Are you able to see my screen? Hello?
Yes, sir.
Yes, sir. Yes, sir.
Am I audible?
Yes, sir.
Yeah. So
Good afternoon, friends. First of all, I would like to thank my company management for giving me the permission to participate in this conference. And also, I would like to thank the management of Orca Jain University and also the organizers of this conference for inviting me for this conference to present my thoughts on this particular subject. And thank you once again. And my session happens to be the last session of the day, in particular during this lunchtime. You know, this is the typical Indian meal time. So it's my responsibility to keep all the attendees awake and attentive to this program. Hope I will do the justice for that. About a month back, when I was approached by the university to present my views, on this particular topic with the theme of novel, natural and novel pharmaceutical research, just I had a reality check whether I am the right person to speak in this conference, because definitely I am not a subject matter expert in this particular subject. But still, looking at my experience, I started my career as a scientist, predominantly involved in herbal research. I spent there for about 8 years. Subsequently, I migrated to the quality management side. In the quality management side, I am taking care of more of modern medicines. So keeping this experience together, I thought I can present my thoughts and viewpoints representing a quality personnel. And when I'm saying quality personnel, I mean that I'm going to present the views of a patient, what a patient can expect in the research side. This is what is going to form the basis of my presentation. These are the discussion topics for the next 30 minutes. We shall start this presentation with few words about research. First, this is mainly for getting the terminology right in our understanding. And we are going to see why we need modern research on herbal medicines. Then we shall see certain approaches which are currently being followed for modern research on herbal medicines. Then finally, we are going to conclude this session with certain challenges what we are encountering in the modern research on herbal medicines. Let's get started with understanding the word research. Basically, in the layman's understanding, there are three types of research. The first one is called discovery. That means it's a process or act of finding something that has already existed. The best example is law of gravity. The gravity was existing even before Newton, but based on his observation of an apple falling from the tree, he did the research and he discovered this gravity. This is one type of research called discovery. Here, it's more about revealing something which is already existing. The second type is called invention. It is an act creating something that had not existed. The example what I have taken is the telephone. The Graham Bell explored the possibility of connecting two persons in different locations being connected by a cable. That's how telephone came into existence. This is a typical example for invention. The third type is called innovation. It's the action or process of making changes in something to improve it. The example what I have taken is the mobile phone. The mobile phone has evolved from the telephone. Basically, the objective is same. Only the thing is they have used a different technology to connect the people in different locations without any kind of cable connection. This is how the research happens, either in the form of discovery, invention, or innovation. I thought of starting this session with this basic explanation because it's very much relevant to our topic, modern research on herbal medicines. We shall be able to correlate these definitions while we are going through the slides. Now let's get into the next topic, why we need modern research on herbal medicines. Animals and plants are the most sophisticated chemical factories synthesizing n number of molecules. Every function in the body, either physical or emotional, has chemical basis. What I mean to say through this slide is, there are n number of molecules available in our body catering to the functions of the body, then definitely we can qualify this body, either it's a plant from the plant kingdom or from the animal kingdom. Definitely they can contribute for evolving the medicines which can contribute to the benefit of the mankind. Herbal medicines, particularly traditional medicines, have been in existence for thousands of years. Let's take the example of our country, India. Here we have got many traditional medicines, starting from Ayurveda, Unani, Siddha, and homeopathy. All these traditional medicines are being in the practice for the last more than 3,000 years. When something is existing for more than 3,000 years, 5,000 years, definitely there must be some substance into that. Without that, they cannot exist for such long period of time. Through accident, trial and error, and through intelligent observation and inference, mankind has accumulated knowledge about useful plants with curative powers. When you're looking at the history, human beings, they have identified a lot of medicinal plants based on the observation of animals. Based on the observation of the animal behaviors, they were able to find out that what could be the curative power of different plants based on their behaviors. In this way, based on the observations through the accident trials, they have accumulated a lot of knowledge, which forms the basis of the traditional medicines. The traditional medicines have survived till today and continue to play a dominant role in the primary healthcare in many developing countries. As per WHO, more than 70% of the drugs used in the traditional medicines are of herbal medicines, and they are contributing to 70 to 80% of the primary healthcare in developing or underdeveloped countries. Now let us look at the other side. Let us look at the typical process for the drug discovery in the modern times. Drug discovery and development remains a lengthy process with low rate of success and huge capital investment. A study in 2020 estimated that the median cost of getting a new drug into the market was $985 million, and the average cost was about $1.3 billion. And also it takes more than 10 years for a new drug to complete the journey from initial discovery to the marketplace. And that too, 1 or 2 in 10,000 molecules are able to hit the market. Now we will be able to imagine how much of work is involved, how much of investment is involved, how much time is involved for discovering a new drug in an existing method of, typical method of drug discovery in the modern medicine. Now being in use in traditional medicines, the knowledge and safety of herbal medicines are relatively better now when compared with the unknown synthetic molecules. This indirectly helps to increase the success rate of drug discovery process and to reach the market faster. This results in reducing the project cost on the other side. Modern medicine has evolved from traditional medicines. When we are looking at the history, we are finding it really very difficult to identify the time of separation between the two. When the modern medicine came into existence, it's really very difficult to find the time period. So modern medicines, I can say from the literatures, I can say that modern medicine started from the traditional medicines, then continued with the synthetic compounds, and in the recent times up to the lot of biologicals. Just we can say the example of Settener isolated, a German scientist, he isolated morphine in pure form from the opium, the first alkaloid ever to be isolated from the plant. The literature says that that could be the beginning of the modern medicine. Again, that invention is based on, again, the discovery was based on certain mythological basis, based on the traditional medicines. Now let us look at the research on traditional medicine, the focus areas. What will be the focus areas for any research on herbal medicines? It is focusing on patient safety, product efficacy, and product quality. Looking at this, let us find out the various approaches being adopted for the modern research on herbal medicines. So based on the literature review, based on my understanding, I have classified the approaches into 4 types. The first one is disease-oriented approach. This approach is mainly applied in disease conditions for which established therapy are not available. Even the modern medicine today, there are many diseases for which we don't have established therapy for that. And we are having a sort of management therapy in all of the situations. This could be the approach. We can find out a new medicine from the traditional medicines. And this is also known as reverse pharmacology by some experts. The objective is to understand the mechanism of action and to optimize safety, efficacy, and the acceptability of wearable products using relevant science. Based on the reference from traditional medicines, the medicinal plants are processed in prescribed procedures when we are adopting this approach. Then such medicines are being evaluated for safety and efficacy with application of modern science using preclinical and clinical studies. Now, many products are available in the Indian market based on this approach for the treatment of chronic illnesses like diabetes, arthritis, etc. Now, in India, we have done a commendable job by integrating the institutions with the industries. Their institutions are working for establishing a standardized products for the industries. And by this way, there are many premier institutes who have done a great job, like ICMR. They have come out with medicines with complete evidence about its safety and efficacy, and such kind of products are already available in the market. The next approach is called technology-oriented approach. Here we are using advances in technology for discovery of new products from traditional medicines. Based on the reference from the traditional medicines, the medicinal plants are chosen for research, screened for biological activity in a series of high throughput screen. And it's aimed to identify active compounds or interesting molecules which is then subjected to classical pathway followed for drug discovery. Typically, this follows this three-step sequence. First step is about pharmacological screening of medicinal plants, then bioassay guided fractionation, then finally isolation of the active constituents. Once the components are identified and isolated, then it's being subjected for structural activity relationship for improved versions of the molecule. Nowadays, we have got advanced computational tools, in silico methods, which enable high throughput screening of genetic information, the prediction of chemical structures, and pharmacological activities based on the huge database. Fortunately, we are having a database for compiling all the research outcome from different researchers across the world, and this database helps us to do many activities without practically doing the physical activities. Based on literature survey and the database search itself, we shall be able to do a lot of screening activity to shortlist the molecules. We've got many success stories applying this technology-oriented approach. I have given the reference of few major discoveries, what we got applying this approach. Though all these molecules have been originally identified from the plant kingdom, many of them are being synthesized now in the chemical laboratories, but still the identification of those molecules from the plant and subsequent research onto its pharmacological activities brought a big difference in the development of modern medicine. Just I wanted to give one example of technology-oriented approach. I have taken the example of artemisinin. Artemisia annua. It's a plant used in traditional Chinese medicine for the treatment of intermittent fever, exhaustion due to heat or fever, lingering heat in joints and bones. Some researcher, he collaborated this particular observation with the symptoms of malaria. Then he started studying on this particular plant. And finally, he has done cold water extraction and fractionation. That became the beginning of this invention of this particular molecule. During this process, initially this particular species of this plant, it was screened for the activity and it was dropped from the research because they were following the typical process of the extraction process using the solvents and also using some hot process for the extraction of this particular plant. So they identified that there was no activity in the treatment of malaria. They dropped it. Then finally, another researcher, he keenly observed that extraction process followed in the traditional medicines. There they followed the cold water for the extraction of the plant. Then again, he resumed the research and he did the cold water extraction, which brought this active ingredient out of the particular plant, and further fractionation led to the discovery, led to the invention of this molecule artemisinin. When the artemisinin was invented, they identified that that molecule is having the availability of only 8 to 10%. that became the limiting factor for the extensive use of this particular molecule. Then subsequently, they have subjected this molecule to the detailed structure activity relationship. They have done a lot of chemical derivatives and landed up in artemether, artesunate, and dihydroartemisinin. These are various molecules which are the chemical derivatives of artemisinin with higher bioavailability. So these are the molecules which are currently available for use in the treatment of malaria, even in the treatment of drug-resistant malaria in various parts of the world. And even many pharmacopoeias are giving the monograph for these molecules. Just I'm correlating whatever we have seen in this technology-based approach to the explanation what we had on the research. It started with the discovery of the knowledge of traditional medicines, Artemisia annua, for the use of the treatment of malaria. Then it led to the isolation fraction of the active principles. There is artemisinin, there is an invention. Then finally, it led to the standardization and presentation of the medicinal product in the form of artemether and artesunate, which are having better pharmacological activity and safety profile when compared to the artemisinin. So by this way, we shall be able to get the best out of this medicinal herbs. The next approach is called standardization approach. This is aimed to develop standards for herbs and herbal products. In our typical language in the industry, we call it as specifications and method of analysis. Quality control of the herbs and herbal formulations is important for acquiring the optimum therapeutic effect as well as for expanding global outreach. When I'm saying that this is very useful for expanding the global outreach, there's always a communication gap between the people from the eastern side of the world and the western side of the world. Whatever we are talking about the value of herbal medicines, they are not being validated in understanding of the western people. By this way, we are not able to get these medicinal plants across this world. So here this quality control approach is going to be a very important tool for standardizing these herbal medicines, thereby expanding the global outreach. And a lot of research has been done in this particular direction. The best example what we can have is Indian Herbal Pharmacopoeia. There we have got so many monographs for the herbs and herbal preparations. And in all this standardization approach, mainly the fingerprinting and marker compound analysis are often used for the standardization of the herbal medicines. Both qualitative as well as the quantitative analysis, they were done using modern instrumental methods of analysis. And marker compounds, whatever they have identified, they may be therapeutically active, in some cases may not be active, but they are helpful for standardizing the particular herbal or herbal preparation. Interestingly, Indian Pharmacopoeia has established standards for more than 165 herbs or herbal extracts or herbal preparations. By this way, we can say that all these herbal medicines are being treated at par with any modern medicines. And phytochemical reference substance and botanical reference substance are also available now for standardizing all these herbal products. And this is a very important aspect for establishing phytopharmaceuticals in India. About 3 to 4 years back, our government, Indian government, has come out with the Gazette notification for introducing phytopharmaceuticals in the Indian market. They have defined what are the requirements for establishing a phytopharmaceutical medicinal product in the market at par with any modern pharmaceutical product. They have defined the pharmaceutical drug as a drug of purified and standardized fraction, assessed qualitatively and quantitatively with defined minimum 4 bioactive or phytochemical compounds of an extract of a medicinal plant or its part for internal or external use on human beings or animals for diagnosis, treatment, mitigation, or prevention of any disease or disorder, but does not include drug administered through parenteral route. Though it's a bit complicated definition, what they want to mean is we can develop phytopharmaceutical drug with established marker compounds. They are saying that minimum 4 bioactive components has to be established for every phytopharmaceutical product in such a way that the whole sum of the plant is being extracted and they are being used in the treatment of the diseases based on the leads from the traditional medicine. And many of the industries are into this particular process now to establish the product, starting from taking the leads from the traditional medicines up to the level of developing the formulations, standardizing it, and finally going to the level of preclinical and clinical studies for establishing safety and efficacy of the product. And at some point of time, they will file it with the Drug Control General of India to get the approval as a phytopharmaceutical drug, which can be sold in the market at par with any modern medicine. And it's also the interesting aspect, important aspect for establishing functional foods to promote better health, to prevent the chronic illness or lifestyle diseases. And nowadays, we have got the regulation which permits herbal-based preparation to be used as a food under various categories like specialty food containing plant or botanicals, or food for special medical purpose, or food for special dietary use. Again, taking the leads from the traditional medicines, we can use certain plants for improving the health conditions of the people. And the fourth approach is called conceptual approach. Here it is aimed to deep dive into the concept of traditional medicines. Here it is understanding more about the traditional medicines. Though the traditional medicines are documented, the adequacy of the documentation is always debatable. So in this case, we are further studying this conceptually. We are studying this conceptually to understand better about these traditional medicines and to create more and more documentation to develop the evidence for the pharmacological activity as well as the safety for the respective products. And it evaluates the traditional medicines and their formulations on the basis of their own healing concepts and principles. And the ultimate objective is to provide evidence supporting concept and therapy. Now let us look at certain challenges what we have in applying the modern principles in the research of herbal medicines. Traditional medicines have setback with respect to the experimental verifications and statistical validations expected in modern medicines. This is the main reason which has caused a big gap between the traditional medicines and the modern medicines. Because modern medicines requires the evidence for each and everything, whether it's in terms of safety or in terms of the efficacy, and such kind of evidence has to be validated in terms of experimental verifications as well as it has to be made statistically valid to prove that they are all safe and effective. There is a place where traditional medicines are not able to give adequate support to qualify for their requirements. Herbal medicines are believed to have built-in safeguards which are difficult to follow in the drug discovery. The example what I have taken is the ephedrine. The ephedrine was isolated from the plant ephedra, but it was found that administration of active ingredient ephedrine for the treatment of asthma caused increase in BP, while the administration of the plant drug did not cause that adverse effect of increasing the BP. Our understanding is the herbal material contains some built-in safeguards which are antagonizing the undesired properties of that particular plant. By this way, the plant medicines are giving better desired results than the isolated compounds, and all these areas are remaining mystery for many of the plants. Traditional medicines includes a large number of plant materials and minerals which pose challenge to researchers. For an example, the well-known Chyawanprash is formulated with around 50 ingredients. And really, the researchers are clueless to find out what all these individual ingredients are contributing for establishing immunity in the human beings. And there are some studies which says that even the 13 ingredients are enough for providing the desired results of Chyawanprash, but still, when we are countering such kind of conceptual understanding of the traditional medicines, we have to be very careful in understanding that, and we have to substantiate with a lot of facts and figures. Traditional medicine often involves certain processing of the plant materials which could not be understood by modern researchers. This process renders the medicines less effective, less toxic, or therapeutically more active. We have seen many terminologies used in the traditional medicines. For an example, the word shodhana, it's a purification process in Ayurveda that's being applied for the preparation of certain traditional medicinal preparations. There they are treating the plant materials through a series of processes with a lot of herbal extracts. And by this process, they are claiming that it is improving the therapeutic efficacy of the product as well as it is also reducing the undesired properties of the plant. But still all such kind of purification process remains beyond the comprehension of the modern researchers. And one more constraint is high level of variability in the research outcome. This could be attributed to inherent variation in the plant source. It could be a species variation or seasonal variations, etc. Research methodologies, inadequate understanding of the traditional medicines. When we are going through the literatures, we can find a lot of literatures supporting the use of, demonstrating the use of traditional medicines in the treatment of many diseases. In the same way, we can also find a lot of researches which disproves that all such plants are not really useful and the results are not really significant when compared to the placebo effect. So when we are having these kind of studies, we are not able to prove it adequately that the herbal medicines are really providing the consistent results. And again, there's a lot of gap between such kind of researches. By this way, we are not able to clearly understand what is the difference in the methodologies they adopted, whether it is a variability from the medicine or it is because of the research methodologies adopted by the different group of people. The lack of proper coordination, communication between the custodians of traditional medicines and modern researchers. There has been always a gap between these two group of people, the people who are doing the research on traditional medicines and the people who are doing the typical process for the drug discovery. The communication is lacking between these two people. By this way, we are not able to get the best out of these traditional medicines. And that becomes a big barrier between these two groups. And that becomes a limiting factor for getting more and more drug leads from the herbal medicines. And ecological consideration for endangered plant species. Nowadays, a lot of plants are being endangered and they are being regulated by the government to protect those kind of species. That also becomes a barrier for doing such kind of research. And there's a lot of social and cultural barriers. We can say that a lot of debate going on, saying them that Eastern system of medicine is better or Western system of medicine is better, and Western system of medicine is having more efficacy, and Eastern system of medicines are far more safer than the modern medicines. There's a lot of debate going on whether East is better or West is better. Why can't we think East and West is the best? If we're able to integrate both the sides, I think we can get the best out of both the systems. Just I would like to conclude this presentation with the words from Rig Veda. Let noble thoughts come to us from all directions. That means, in this context, what we discussed, let us accept, respect, and test the validity of all the knowledge that is available, in particular in the area of herbal medicines. Thank you so much. Due to the constraint of time, I have rushed a bit to complete it in time. Thank you.
Thank you, sir, for sharing your knowledge with us. We are very privileged to have you for today's plenary session. Now, may I request to Mr. Sumanta Singh, Associate Professor, School of Pharmacy, Arkajan University, to kindly felicitate Mr. K. Shravana for his contribution in this plenary session.
Thank you, sir, for your wonderful presentation. Please accept this memento from our side.
So,
thank you so much, sir, for this nice presentation. Please accept this token of appreciation from our end.
Thank you, sir. Thank you so much. It's my privilege to participate in this e-conference, to be amidst with you people.
Yes, sir. It was a nice session that was enlightened on the research on natural products. We know there is many challenges which for that the Ayurvedic product or our natural medicines, they are struggling. And whatever you enlightened with your knowledge, really it will help for the budding scientist to overcome those challenges. Thank you once again.
Thank you.
कौन-कौन जो छात्रों का नाम है
Thank you, sir. Now, this brings us to the end of today's plenary session. May I request to Mr. Sumanta Sen, Associate Professor, School of Pharmacy, Arkajan University, to kindly give vote of thanks. Sir, please.
Good afternoon to all of you. Once again, that is this 3 days e-conference, which the topic is natural and novel pharmaceutical research. First of all, I like to congratulate and salute to both of our Dean, Madam Fatima Tezro, and our beloved Dr. Jyotirmay Sahu for organizing such an event that is not only beneficial for the faculty members, the students, the other peoples who are join this conference. And I hope the topic, which is based on the natural products, we know the severe challenges is there to develop any sort of natural products, but our speakers, Dr. Krishna Priya Mohanraj, Dr. Ajay Simalti, Professor Fatima May Tezro, and Mr. Sarvan Kumar. They are very senior persons who are working since last few years. And we are really, that is not only the students, the budding scientists, we can understand the challenges which shared by our distinguished guests. And first of all, I want to congratulate both the universities, that is Arkajan University from Jharkhand, India, and Pharmacy Department, St. Pedro College, Philippines. And lastly, that is the students. The students, I know that is a few years back, I was also a student. We can say that is a blessings of this COVID situation. That's why it was quite easy. All the peoples come in contact with each other through these conferences, which was quite difficult before of that. But it is a blessing of, we can say that is a blessing of COVID-19. A number of technology we adopted, and for this we come in contact with each other and getting together to share our knowledges. And we know that sharing the knowledge increases the efficiency, it can also change the working ability. Once again, I not wants to lengthening this vote of thanks. Once again, I wants to salute both the deans from both the universities and colleges who that is organizes such a wonderful events. And once again, I wants to congratulate the coordinators, the students who make their that is for their precious time and patience. Thanks to all of you once again.
Thank you, sir. Now, I would request to everyone to please stand up for national anthem.
Lupa गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you everyone for your presence. Now we will meet for scientific session after a lunch break at 3:20 p.m. Thank you.